gentlemen, dear jury members, dear young scientists, welcome, bienvenue, welcome to the second day of the 51st edition of the National Jonk Fuerscher Contest.
A quick recap of a day. We have 43 national teams from 15 different schools and universities.
The 51st national contest is being held under a hybrid form, which means that each team gets the opportunity to come on stage to present their project in front of the jury while being filmed for the live stream. However, this edition also marks a comeback to the traditional format, because as of tomorrow noon, you will be able to meet the participants and discover their projects, set in a wide range of scientific fields here at the forum. The open house will be followed by the award ceremony at 4 p.m. Don't miss the opportunity to get to know our young scientists and ask them questions about their projects.
We now have the honor to welcome on stage Mr. Serge Coisotti, the president of a jury, who will officially open the second day of events.
Ladies and gentlemen, dear young scientists, dear Mr. President, dear Carlo, dear Ms. Director, dear Susanna, dear colleagues from the Young Scientists Foundation team, dear colleagues, jury members. It's my great pleasure and honor to have once again the privilege to open the second day of the Young Scientists Contest 2022 in my capacity as the chairman of the jury of the contest. A jury composed of distinguished experts from our Luxembourgish science, technology, and research world. Welcome and thank you all for taking the time to be here today. For 6 years now, I have had the pleasant opportunity to interact with many of the most inventive and talented young people of this country. Every year, our young scientists keep surprising myself and my colleagues from the jury with their ideas, their tenacity, and above all, their creativity. This year again, it is no different. Over the years, we have seen the foundation and the national contest grow and evolve. We have seen many participants come back year after year with new or renewed projects, determined to make their ideas happen and to show them to the world. We have also seen many new participants encouraged and motivated by the success stories of their classmates, doing incredible work and receiving well-deserved validation for it. Since 2017, many things have changed, and it's extremely encouraging to see the improvements which were made since then. We have had a steady increase in participants and projects. The quality of presentations and projects improved thanks to the support from schools, coaches, and field experts. And finally, the Foundation's most recent initiative, the Science Next program, shows enormous potential. This extracurricular initiative for young scientists was created in 2021, and it consists of a series of workshops that are partly animated by my colleagues from the jury. These workshops address different topics, ranging from ideation and mind mapping, how to improve one's presentation skills, to how to conduct correct information searches by using patent information, and much more. I am personally convinced that this program provides many valuable skills to the participants that they can apply to the contest, but more importantly to their future academic and professional careers. I want to congratulate the Young Scientists Foundation for this excellent initiative. The national contest, which is the origin of everything we see here today, has evolved quite a bit since it took its first steps. It is no longer just a science competition. It is also a platform where young scientists can exchange ideas and interests with other like-minded people, other young scientists and field professionals like the members of our jury, for example. The role of the jury is not only to evaluate project papers and distribute awards, but also to encourage and guide the participants in their scientific journey. We support young scientists by helping them to develop their projects, their knowledge and skills, thus bringing them to the next level. On that note, I would like to thank all my esteemed colleagues from the jury for their devotion and their readiness to transfer their own passion for science to the next generation. Last but not least, I would like to address our young scientists. Despite the difficulties and restrictions of the last couple of years, you have never faltered, never given up, and never lost your motivation and ambition. You still created, researched, participated in the contest, and showed that nothing can stop you if you put your mind to it. And for this, I applaud you. Let me now wish you all a pleasant and inspirational second day of the Young Scientists Contest. Thank you.
We now welcome on stage our first participant of the day, Sky Beck from the International School of Luxembourg, who will present a study on the effects that a diet has on liver cancer and angiogenesis. Welcome, Sky.
Good morning, honorable members of the jury. Cancer is something that almost everyone knows about, and certainly it is something that we all know might happen to us as we grow older. However, only 5% of cancers are genetic, meaning the other 95% come from environmental factors, many of which come from diet. My name is Sky Beck, and today I will be showing you some of the science behind how diet can significantly reduce your risk of developing cancer throughout your lifetime. So let's start with the basics. When your body has a small cancer tumor, that tumor will only be able to grow to a certain size until it runs out of the resources it needs, like oxygen, food, and nutrients. Therefore, some cancers use a super important process called angiogenesis. This is when it forces the blood vessels in your body to grow in and around the cancer tumor so that it can have the oxygen and the resources it needs. And now that the tumor has angiogenesis, it can grow exponentially. See, if the tumor does not have angiogenesis, it will stay small. So this means that we basically all have some forms of cancer in our bodies at the moment, which will never become harmful to us because they will never have angiogenesis to get what they need. On top of this, when cancer gets big enough, some of the tumor cells can leave the tumor and go into the bloodstream, where they can be transported anywhere else in the body and create secondary tumors. Unfortunately, it is at this later stage of cancer that most are detected and diagnosed, which is what makes cancer so dangerous and so serious. Now, in fact, a study from Dr. Lee found that when angiogenesis is out of balance in the body, it is the root of over 70 major diseases affecting over 1 billion people in the world today. And this is huge, since this is only a fairly recent discovery, and so there's not a lot of data on it. So I asked myself, well, what if we could just prevent angiogenesis from getting so out of control in the first place? And it turns out we can, with what we eat, which allowed me to formulate the following research question. To what extent does diet positively or negatively affect the processes of angiogenesis and subsequent cancer? Now, one of the ways angiogenesis can be suppressed is within our control through diet, which causes around 35% of cancers. So I researched 3 dietary factors that I thought were fairly interesting. These were resveratrol, something that you find in red wine, genistein, something that you find in soy, and ellagic acid, something that we find in almost all berries. And this is what I found. So, as you can see, I put the chemical formulas for the 3 dietary factors I chose on the screen to the right. And what they all have in common are these hexagon-looking shapes on them called phenol functioning groups, which I highlighted for you in yellow. Now, the role of these phenol functional groups can be seen in this picture that I made for you. Now, on the right side, the phenol functional group enters your bloodstream, and it causes a reduction in PKG1 kinases. These are the proteins that are responsible for the process of angiogenesis. See, on the left side, where there are no dietary factors, in this case resveratrol, there was a higher level of PKG1 proteins, and therefore a higher level of angiogenesis, which you can see from more blood vessels. Now, my research focused particularly on liver cancer. However, the data showed that this is applicable to nearly all cancers in general. Now, this is super, super cool because it means that we can significantly reduce cancer on our own accord. We can even mix dietary factors together to have a higher suppression of angiogenesis. In this case, curcumin and genistein were mixed, and they were higher at inhibiting angiogenesis than the 2 dietary factors alone. On top of that, Dr. Lee found that some of these dietary factors are more effective at inhibiting angiogenesis than some prescribed medical drugs dedicated to combating cancer. Now, unfortunately, while diet can have a positive effect, it can also have the inverse effect if done wrong. The 21st century has seen the highest levels of obesity worldwide. When eating a high-calorie, high-fat diet, all the fat, excess fat, has to be stored in the body, and angiogenesis plays a big part on how this is done. Fat reserves need their own blood supply, and your body uses angiogenesis to do this and store all the excess fat, which can lead to cancer in the future. As you can see here, the increased fat forced more blood vessels to grow. Now, I took a closer look at the United States because it had the most data available on obesity, cancer, and angiogenesis. And some of what I found made sense with the data, that there was a correlation between obesity and cancer rates in the US subsequent from angiogenesis. Now, at this point, I am just completely scratching the surface because on deeper inspection, it leads to a whole field of epigenetics and gene expression, and how dietary factors such as energy density and cultural diets can affect angiogenesis. And this is completely revolutionizing how everyone is looking at cancer all around the world. The lack of data in this field presently is not allowing me to answer my research question with certainty at the moment, but it is what has drawn me to the fields of medical research and science, because at the moment, this field has almost endless possibilities for future work. Thank you very much for listening. I'm open to any questions you may have.
It's now time for a video of our former Jonk Fuerscher, who wanted to share some words of encouragement with our current participants.
Hey, my name's Valentine. I participated in the Jonk Fuerscher National Contest in 2017 and 18 with 2 friends of mine on a project about a concrete 3D printer. I'll be helping during the contest, so if you see me around and need help about anything or have any questions really, let me know. And then I'll wish you guys good luck. Make sure to enjoy the time you have here, and I might see you guys around the contest then.
Hi, my name is Marie and I've participated in the Jonk Fuerscher twice before. It's had a huge impact on my life and what I'm studying now in university. So this year I'm really excited to meet you guys and learn about your projects. Good luck on the presentations on stage, and I hope you have a great time and enjoy the rest of the contest.
Hi everyone, my name is Songmin Park and I'm an alumni of the Jonk Fuerscher Youth Science Competition where I participated last year and the year before that. Through my experience with the foundation, I was able to meet so many cool new people from all around the world with the same love and passion for science. And I also got to develop my own interest in the subject and its disciplines. And I'm currently now studying engineering science at the Technical University of Munich. I just wanted to come along and say a huge congratulations to everyone who participated this year, because it definitely must have been a challenge with the current global circumstances. But I mean, that's the beauty of science, isn't it? When we are faced with a challenge, we come up with new, creative, and innovative solutions to tackle it, and we learn more about the world and also about ourselves in the process. So good luck to everyone and congratulations.
Hello, we are Almina, Katrin and Anna. Last year we participated at the Jonk Fuerscher contest, and this year we will be helping as volunteers. We are really excited about your project and are looking forward to meeting you. Good luck, enjoy your presentation and have fun.
Hey Jonk Fuerscher participants, my name's Alex Cowan. I'm an alumni of the contest and I'm currently on campus at the University of Queensland in Australia, where I'm studying a Bachelor of Chemical and Material Engineering. You see that over there? That's our new chemical engineering building, that glass thing. It's awesome. I just wanted to commend you guys for putting in the effort to participate in this year's edition of the Jonk Fuerscher contest, because it's one of the best things you can do at this age to get in contact with the scientific community. It's the first step into all of this, where you meet people who are like you, who think in the same way you do, and everything. It's just fantastic. You feel like you're where you're meant to be. So this is where I'm meant to be. Where could the Jonk Fuerscher lead you?
Our next participants are Selena El Hafidi, Sophia El Hafidi, and Shireen El Hafidi from the Lycée Michel Lucius. They all agree on one thing. Apples possess a strong immune system booster. Let's hear more on their project called How Apples Keep the Doctor Away.
Good morning. If I tell you available worldwide, cheap, and a lot of varieties, you might think about
apples. Have you ever heard about the sentence, an apple a day keeps the doctor away? Is it true?
But what makes them so healthy?
Oh, this is because of quercetin. Quercetin has a neuroprotective effect. It helps to prevent age-related, and it protects the cell.
There's not just quercetin in apple, there's also fiber. Fiber can reduce the level of cholesterol in your blood, but also reduce the chance to death from a heart disease, and can make your digestive system healthy.
Let's not forget about the most important component of apple, vitamin. Because yes, apple can contain a high quantity of vitamin A, B, and C. You probably wonder, what's that effect in apple? Well, vitamin can boost your body resistance and protect your immune system.
So, as you know, component of apple can help people to fight against some diseases, like cardiovascular diseases. Cardiovascular disease cause the most of death in the world because it affects your heart and your blood vessel. But how does it work?
We all produce a certain amount of cholesterol in our blood for the simple reason that we need cholesterol to produce healthy cells. But having a high level of cholesterol recruits fatty deposit, and this deposit will grow, make it hard for the blood to flow. And this can really take big proportions because there will not be enough oxygen carrying by blood cell reaching the heart.
But how eating apples in our alimentation can keep the level of cholesterol healthy? So having a rich polyphenol diet can relax your vessel and produce nitric oxide. Polyphenol in apple can also help managing blood pressure level and promote a good circulation of your blood.
How to use the benefit of apple to fight against acne. So acne can be caused by free radicals. Free radicals come from environment factors like pollution, smoke, or other chemicals.
Free radicals are unstable molecules. They're missing an electron. So they need to steal an electron from a stable molecule in order to balance them out. And this process is called oxidation.
Oxidation can cause a lot of damage, like oxidation stress. It's when the cell becomes too weak, so it drives the acne process and let bacteria enter in it.
Antioxidant is a solution against free radicals because it can give an electron to the free radicals without being unstable. Apple peels contain a lot of antioxidants like quercetin, flavonoids, but the most important one is vitamin C.
So we did a little experiment because we wanted to know how we can reduce acne. How acne is produced? Well, is stable molecules bind with free radicals, and this causes acne, but apple can stop that.
So we cut a peel of apple, so the antioxidant in it can kill all the free radical, and we put a thin layer of honey, so honey can remove all the dead cell caused by free radical. We did that for 3 days and 15 minutes.
So here we can see the results of our experiments. The first day, the acne is really huge. Then the second day, this picture shows us that the acne has decreased. Finally, the third day, we can see that the acne has mostly disappeared. And the final results can show us that the acne doesn't exist anymore.
So by this experiment, we can really see that apples can keep the doctor away. And by this presentation, there's a lot of benefits in our organisms. We really want to thank our parent who has been a big support, and also our mother for the experiment. And thank you.
Thank you.
Thank you.
Let's have a little exchange with Sky Beck to know about his motivation to participate in the National Jonk Fuerscher contest.
Good morning, Sky.
Good morning.
Welcome, and thank you for being the courageous first one today. We have a few questions to ask. We're curious about hearing your answers. So, first one would be, do you implement your findings regarding diet and lifestyle into your own life these days?
Well, my research really shows that it's more the older you get, the more prone you are to spontaneous cancers. For example, basically everyone over the age of 70 years old will have some form of cancer in and around their throats. And these will be really tiny, tiny cancers, like 0.5 millimeter in volume. And so they're going to be really small, and they're not going to affect a lot of people. But my research really goes into detail on how you can just reduce that, and how those cancers anywhere in your body will never be harmful to you. And so, to answer your question, at the moment, as a younger person, I probably won't have to worry about some of this more than someone who might be a little bit older. But it's definitely something that I'm now conscious and aware of, that I can now use moving forward and make healthier choices, especially when it comes to obesity and a higher fat diet.
Sure. And maybe consult the elder generations on that. Yeah.
Also, we were curious about your future. Do you plan to pursue studies in biology or medicine in the future?
Definitely, yes. I've applied to medicine in Ireland, and I've had to take the HPAT. No response yet, because that takes a while. I've also applied for biochemistry and chemistry in Canada, which is where I'm from. And so far I did get an admission into McGill for chemistry.
Oh, wow.
So I'm very happy about that. But yes, I'm definitely going down the path of science or medical science, definitely.
Yeah, interesting to hear that. Good then. When doing your research, your project, was there any surprises coming up you didn't expect before starting that project?
Yes. When I was a child, I thought that most cancers were genetic. So I'd say like most cancers were genetic. And to be honest, at some point I even thought that all cancers were genetic. But my research immediately proved me wrong, because only 5% are genetic, and the other 95 are just random environmental causes that you can be exposed to, like diet, alcohol, hepatitis, and stuff like that. And so something else that surprised me was how angiogenesis plays a big part in all of this, and how just your blood vessels is the root, the main focus that branches out and affects all these diseases in the world, 70 diseases affecting 1 billion people in the world. And this was super surprising to me, because scientists always used to look at these individual diseases like cancer and obesity individually, on how to cure them and how to tackle them like that. However, now everyone's perception is completely changing because now scientists and communities are looking at a way to go at the roots, which is angiogenesis, which can prevent so many of these diseases that are affecting so many people.
Right. Would that be also a topic you would like to follow in your studies?
Most definitely. I think McGill could have a lot of resources that I could use. But then again, this, as I said, this is a fairly recent discovery. This is fairly new. There's not a lot of data on it. It's only now changing how we're looking at cancer and all of these diseases. So there isn't a lot of data out there that is able to move forward and to be able to research. Obviously, there are so many factors that affect these diseases, like cancer, and not all of them are diet. Like I said, there's alcohol, there's hepatitis, and so there's so many combinations that we can put together to see how it affects angiogenesis. So that, yeah, there's just so much research. There's almost endless future work in this topic that I'm definitely willing to explore.
Yeah. And maybe a final one. When doing your research, did you also come or look into, or were you interested in looking into how cultural diet plans or habits then have an effect on those diseases? Or could you tell us which place is probably the healthiest in the world looking at diet plans?
Well, I wouldn't be able to tell you which culture is the healthiest, but culture was definitely something that I looked into. I started by choosing the United States because it had the most data. And then I focus more on looking at different cultures and looking at how they eat, how much fat and what their diets contain. And then based off of that, I started comparing some of the cultures there. And that was where most of my data came from. But then on further inspection, what that basically means is that all of these cultures and these diets are in a whole new field of epigenetics and gene expression, which is basically, which changes everything. So it's not really just a culture thing, it's more going into genetics now, which makes everything more complicated. Yeah.
Thank you very much. It was very interesting to learn about, and wishing you all the best for your way ahead.
Thank you so much.
Thank you.
I'm sure that there are a lot of coffee lovers watching this contest right now. Well, this next project might interest them. Please welcome on stage Luisa Gomiero from the International School of Luxembourg, who will present her research on the impact of caffeine on enzyme-catalyzed reactions.
Good morning. How did you wake up this morning? I'm guessing you woke up just like me, feeling tired, and so you looked for coffee or tea just to have some caffeine in your body to wake up. But have you ever wondered what impact that caffeine has on your body and especially the liver? This led me to my research. So this was my research question. To what extent does the production of oxygen affect the reaction of 1% hydrogen peroxide solution with 1 centimeter cubed liver pieces soaked in a range of caffeine concentrations for 24 hours? Now I will explain what this research actually was. So some background, first of all. I took a basic enzyme reaction, which is the catalase reaction, where we have a source of catalase, which either from potatoes or liver, for example, and you add it to hydrogen peroxide. This produces water and oxygen. However, I wanted to investigate how caffeine impacts this reaction. So I had to first of all keep in mind a few things that affect enzyme reactions. The main ones being pH, temperature, and substrate concentration. Thus, keeping the same concentration of hydrogen peroxide, 1%, keeping the same temperature, which was at room temperature, and the same substrate concentration, meaning that whatever source of catalase I used had to be the same amount for each trial. So what happens to the caffeine that is metabolized within your body? Caffeine is metabolized by a cytochrome CYP1A2 and creates 3 different products. The main one being pyroxenine. This is a central nervous system stimulant, so this is what makes you feel awake in the morning. So I had to create all of my controls in order to have sufficient data to draw some conclusion from my experiment. The first one, I had to determine my source of catalase. I tested both potatoes and bovine liver. I soon found out that potatoes didn't produce nearly enough oxygen to be measured. Therefore, I decided to use liver pieces. Next, I determined to use 1% hydrogen peroxide. I also tested 3%, but that produced too much oxygen, and the glass gas syringe could not measure that amount of oxygen. And I used 30 milliliters, which was just enough to have the liver pieces soaked in it. Next, I determined my caffeine concentration, my caffeine mass, and my range, which ranged from 0.02 grams from 0.1 grams. I also had a control trial where I did not use any caffeine. Next, I determined the surface area of my catalase pieces, which was 1 centimeter cubed. And I determined that I would let the liver pieces be in contact with the caffeine for 24 hours to allow the caffeine to be absorbed within the liver pieces. So my methodology. The first step was to measure the various masses of caffeine from 0.02 grams to 0.1 grams, place them in 100 centimeters cubed of distilled water, and allow it to dissolve, and I use a magnetic rod to aid this process. Next, I cut the liver pieces in 1 centimeter cubed as precisely as I could, which was very challenging, and I placed them in the caffeine solutions previously made. I put 5 pieces of liver in each of the 5 different concentrations. Then, these were left to soak for 24 hours. In the following 24 hours, I measured 30 centimeters cubed of 1% hydrogen peroxide, placed this in a conical flask, and I set up my glass gas syringe with a tube and bung. Next, I placed the liver piece in the conical flask with the hydrogen peroxide, closed the bung, and the oxygen produced was collected in the glass gas syringe. I recorded my data for 2 minutes every 10 seconds, and after 2 minutes, most of the reactions had already finished. So what were my results? So these are my average results for each of the concentrations for the 2 minutes. It might be a lot of data, but there is a clear trend here that for the highest mass of caffeine used, which was 0.1 grams, we can see in green at the top, had the highest total volume of oxygen produced. Whereas my controlled, which can be seen in blue at the bottom, had the least amount of oxygen produced. But I didn't only want to investigate how the total amount of volume was affected, but I also wanted to investigate the rate. The rate is fastest during the first 10 seconds of the reaction, as that is when the greatest amount of oxygen is produced, which I created this graph from. As you can see on the x-axis, as the caffeine used increases, so does the rate of reaction. So it is clear that the caffeine acted as a stimulant within this reaction, increasing the overall reaction. So you're probably wondering, what do these results actually mean? So the first thing, as I said, is that the overall enzyme-substrate reaction had increased its rate. That means that coffee acts as a stimulant within your liver cells and does not inhibit them. Then we can also determine that in hypoxic conditions, which means when oxygen is not present or available in your liver, caffeine has the ability to partially restore this catalase activity within your liver. Overall, this means that the respiration within your cells increases, as well as the caffeine has an impact on your cytochromes. Cytochromes are present in the electron transport chain, which is a step of cellular respiration. Thus, the overall cellular respiration had increased. But these cytochromes are not only present in the electron transport chain within cellular respiration, but also they are present in the process of photosynthesis. So perhaps it could also have an effect on photosynthesis. So there were quite a few limitations, of course, as this research was done in a school lab. But here are some of them, but I think the most significant one that truly could have affected some of the data was the surface area of the liver pieces. The liver pieces have a very slippery nature to them. I don't know if anyone's ever tried to cut liver, but it is very, very challenging. So trying to cut them equally for each trial was probably the major random error within this data. Therefore, for the next data collecting or next experiment, I would have to find a better way to cut these cubes. So there were two uncontrolled variables that could have also systematically affected my data. One, the main one being temperature, as this was an exothermic reaction which could be seen through the condensation occurring in the conical flask. And the overall temperature, as mentioned at the beginning of the presentation, could have affected the overall reaction, and the temperature change could have also affected the pH, thus also could have affecting the reaction. So this project for me was very interesting, but it led me to quite a few questions. Does this trend continue for higher concentrations of caffeine, or will it plateau, or will it in future inhibit even the liver cells, as well as investigating other stimulants which are used on the daily from us humans, such as nicotine? Does it have the same impact than caffeine? As well as exploring the other metabolic pathways that caffeine is involved in, such as digestion, and to see if caffeine has the same impact on every single metabolic pathway. Thank you, and I look forward to answering any questions.
Let's now go back to Selena, Sophia, and Shirin. They will answer some questions and hopefully tell us what got them into science and research.
Bonjour, Selena, Sophia et Shirin. Donc, pour votre projet, d'où l'inspiration vous est venue pour ce projet?
C'est notre grand-père qui a mangé beaucoup de pommes. Et du coup, un jour, on s'est dit, faudrait bien qu'on parle de ça.
Et en plus, on mangeait en fait des pommes un peu tous les jours. Et c'est devenu une sorte d'habitude. Mais on le faisait sans vraiment savoir en fait pourquoi on le faisait. Et du coup, un jour, on s'est dit pourquoi. Et en fait, on était sûr qu'il y avait de la science derrière, qu'il y avait quelque chose de scientifique qui voulait
expliquer.
Voilà.
Les vertus médicinales.
Et à vous, personnellement, quel est votre fruit préféré? Est-ce qu'on peut croire que c'est aussi les pommes?
Ça fait partie de nos fruits préférés.
Encore plus après votre recherche maintenant.
Ça prend plus de sens maintenant.
Très bien. Comment ça s'est passé, votre collaboration dans le projet? C'était harmonieux? Est-ce qu'il y a eu des obstacles aussi entre vous? Comment vous avez fait?
Non, après on est des sœurs, donc on se bat beaucoup. Voilà. Mais voilà, on s'est beaucoup amusé, on a beaucoup ri.
Il faut communiquer. Après on a chacune un point de vue différent et on a chacune l'envie de faire à sa manière. Et du coup, il faut se mettre d'accord, il faut communiquer. Enfin voilà quoi.
Il faut mélanger nos idées en une.
Qui plaise à tout le monde.
Oui. C'est bien. Pour vos études, vous savez déjà ce que vous comptez faire par après?
Dans la science,
c'est sûr.
Continuer dans le même chemin?
Oui, dans la recherche.
Et après cette expérience au concours, vous planifiez une participation pour le prochain concours, pour l'année prochaine?
Oui, on compte le faire ensemble l'année prochaine.
Le concept est vraiment cool.
Il y a en plus, il n'y a rien à perdre, donc c'est une très bonne expérience à faire.
Et vous allez continuer la recherche sur le même sujet ou prendre un autre sujet?
On va changer de sujet.
Non, on va changer de sujet.
Vous avez déjà une idée de quoi comme sujet?
Non, pas encore. Mais on y pense.
On avait parlé
du venin.
Du venin des scorpions pour
aider à guérir l'Alzheimer. J'avais lu un article sur le journal qui parlait de ça. Je me suis dit que ce serait intéressant de montrer les bienfaits du venin.
toujours l'impression de lier les résultats scientifiques avec la réalité, eh bien, la vie de tout le monde. Ok. Bon, pour le moment, on vous remercie pour la participation.
Merci à vous.
On vous souhaite bonne chance pour demain. Et bon, en espérant à bientôt pour l'année prochaine. Alors.
Et toutes nos félicitations déjà pour avoir participé.
Merci.
The next project in our competition is titled A Comparison of Toothpaste at Bacterial Inhibition. The aim of this investigation was to find out which toothpaste would be better at inhibiting bacterial growth. Arvid Andersens will now take the floor.
Hello, My name is Arvid Anderssons, and today, as you've just heard, I'll be presenting to you my investigation. My investigation involved looking at different toothpastes aimed at different demographics to address the issue of childhood caries. I looked at the toothpaste and saw how they compared when inhibiting the growth of Lactobacillus casei, a bacteria. Behind me are a plethora of flags. Now, these flags represent my global and international background. But you know what else has an international and global presence, or even background? Caries. In fact, 52% of children, ages 6 to 11, have caries in their primary teeth in the United States of America. The World Health Organization deems dental caries the most common uncommunicable disease worldwide. But why is this of interest to me? Why am I interested in this issue? And why did I decide to explore it? The answer is really simple. I have an inherent interest in dentistry as a prospective career. And of course, in dental school and beyond, I'll be expected to perform my own research, whether it be passion or business. As such, I saw this opportunity to conduct my own research, my own investigation, as well the perfect opportunity, as I'd be able to not only develop my skills in the lab at an early age, but also get to read scientific journals, analyze them, and increase my knowledge in biology in general. So before proceeding with the method, the results, etc., I'd like to introduce to you the toothpastes that were used. So here's a data table, an extract from my paper. The first two toothpastes are both aimed at the 6+ demographic. The first toothpaste is aimed at the 6+ demographic and contains a lot of green, natural ingredients. The second toothpaste is also aimed at the 6+ demographic, but contains industrial, more traditional ingredients. I thought it would be pretty interesting to compare these two toothpaste aimed at children, one with industrial and one with natural ingredients. The final toothpaste is aimed at the 12+ demographic, so adults in the dental world. I decided to compare it to see if there is a difference in inhibitory qualities from 6+ toothpaste and adult toothpastes. One thing that should be noted is an outstanding material in the third toothpaste, so a differential, is stannous fluoride. This will be an interesting talking point later. So the method can really be broken down into 3 major steps. So step 1 was creating the liquid growth medium, or the broth. So to do this, I followed the parameters on the third box to create the broth in which the bacteria would grow. Once I've done this, I used an inoculation loop to scoop some solid pre-prepared stock of Lactobacillus casei. I then mixed this in a flask and put the flask in an incubator at 35 degrees Celsius to let the bacteria grow. Once I observed no contamination and some growth, which I checked with my supervisor, I knew I was ready to create the agar and the petri dishes. To create the agar, again, I followed the parameters on the bottles. And once the agar was ready, I poured it into the 8 petri dishes. I let it solidify, and then I used a cork borer to create 3 holes. Before creating the 3 holes, of course, I drew a vertical line and a horizontal line on the back of each petri dish, splitting it into 4 quadrants. So 3 of these quadrants would be occupied by each of the toothpastes. The 4th quadrant was a control. There would be no toothpaste. I then put all the petri dishes in an incubator at 35 degrees Celsius. The final step, of course, was to gather the results. Data collection. So I did this by taking a picture of the petri dishes for each day. So here you see day 1. This is the first day of the pictures, and day 5, so the last day of the pictures. Now, you will notice these red dots. This is not the natural color of the colonies. That would be, I guess, pretty concerning. No, these are markings I made with my Surface pen to manually count the bacterial colonies. I did this because there was no clear zone of inhibition, a traditional method in these type of labs. As you can see, this is my raw data. It's pretty intimidating. So the reason for, if you look in the third column, I have M's and S's. And this is because the bacteria were not homologous in their nature. Some of the colonies were big, some of them were small. As such, when I counted them, I made sure to count the amount of medium, big, and small colonies and awarded respective points for each. Medium colonies were worth 4 points, big colonies were worth 16, and small ones were worth 1. I then added this up to find the colony growth score. I then graphed the colony growth score for each of the quadrants after 170 hours of growth, which is the total amount of time. As you can see, some of the data is pretty close. For example, the two toothpaste aimed at the 6+ demographic, PS, Schmelz, and G, green, the one with natural ingredients, were pretty close. One minute. When you factor in the error bars, you would see you needed to perform some statistical test. So I performed a Mann-Whitney-U test for all trials against the control and the 6+ toothpaste. I found that all failed the test, not showing statistical significance, except the control against Oral-B. This made me curious to investigate the other toothpaste against Oral-B to determine if there's really statistical significance. And I found that yes, there is. So the conclusion that can be drawn from this, the only firm conclusion that can be drawn from this, is that the toothpaste aimed at the 12+ demographic, Oral-B, was more successful at inhibiting the growth of Lactobacillus casei than the other two toothpaste aimed at the 6+ demographic. I believe this is a rather good conclusion. As earlier I mentioned, the outstanding material was stannous fluoride. In my dental school and in my future research, I think this can be an incentive or a basis for future exploration to see how stannous fluoride can either be implemented into toothpaste aimed at the 6+ demographic, but weighing the benefits and the risks, or seeing if stannous fluoride was really responsible for this increase in inhibitory quality. Thank you very much.
Please welcome back Luisa Gomiero for a short interview to hear more about her experience as a Jonk Fuerscher so far.
Hello, Luisa.
Hello.
Pleasure to meet you.
Pleasure
Thank you very much for the earlier presentation. Yeah, I would like to ask you some questions. First one would be, obviously, talking about caffeine, coffee is the first thing which comes into people's mind. Are you personally a coffee consumer yourself?
I am personally a coffee consumer. I started very recently, in the past 2 years or so, with high school getting more difficult and getting a lot more work and pressure. Caffeine has definitely, let's say, saved some of the days and definitely been there when I needed help to get that little extra wake up a bit more and get that bit more of energy that I needed. So that's what inspired this experiment of mine, because using it every day, you wonder what impact it has on your body. So.
Sure. And have your consumption habits changed since studying this topic then?
I think they have increased. I don't think they necessarily should have, but I think I have slowly increased and upped a bit the caffeine, but I don't think it was necessarily due to the experiment. It was mostly coincidental, but they have changed a bit.
Sure, yeah.
Where did the inspiration for this project come from?
It came from a previous lab that we had done in school. So we investigated enzyme activity in general. So investigating the three major things that impact it, which are pH, temperature, and substrate concentration. But I wanted to expand it further and see what other things actually impact this enzyme activity. What are things that inhibit the production or stimulate it? And so that's where I got this inspiration. And then I wanted to combine it to something that we use every day. So I managed to combine two fields that had interested me. And so that's what led me to doing this experiment and these results.
Sounds good. Congrats.
Thank you.
Do you feel now after the project that the topic is sufficiently explored, or do you have some points which you actually want to explore further?
There are definitely some points that I want to explore further. One of the main ones is just expanding the range of the caffeine that I test to see if the trend that I determined continues or if it changes, as well as seeing the effect of other stimulants that we use every day, as caffeine is one that is used every day and is considered a stimulant, but so is nicotine, for example. That is something we use every day, and would that have a similar impact on this reaction? Do all stimulants that we use on the daily have the same impact? As well as investigating the actual other metabolic pathways that caffeine is involved in, such as digestion. Would be interesting to see the impact that it has on digestion, and creating an experiment related to that and seeing how it affects it would be interesting. Yeah.
Mm-hmm. Do you already know what you're going to study in the future? Are you going to continue in science-related subjects?
I will definitely continue in science-related subjects, as that has been my interest since I was very young. I am currently going to study at the MSP, Maastricht University, at the science program, where I have the opportunity to do various experiments through the projects session of the year, and that would definitely allow me to explore further this investigation of mine and continue collecting data on this, as I think that there are still various areas that I wanted to research within this caffeine and enzyme area. Yeah.
Maybe a final question now, after your experience here in this contest, is there any advice you can give for students who are interested in participating in this contest for maybe next year then?
I would definitely say to participate, even though me personally am very shy when presenting and I get very nervous, but going ahead and getting out of your comfort zone and presenting all the hard work that you've put into your research is truly amazing, as well as exploring what other people have done is fascinating, and you get insight on so many different fields of science that some you didn't even know existed. So I think it is truly amazing, and everyone should participate to at least broaden their field of science and get more interested within this amazing field.
Thank you very much, and that's really truly motivating words, and thank you so much for being here and giving us some further insights into your research.
Thank you for having me.
Consonance and dissonance are the key building blocks to any form of music. But what exactly are they? Welcome now on stage Daniel Jenkin from Athénée de Luxembourg, who will present his project called An Investigation of Musical Consonance and Dissonance. Please.
Hello everyone, my name is Daniel Jenkin, and today I will present my investigation into musical consonance and dissonance. Now, to start off right away, I will play you two intervals, and you will have to tell me which one sounds nicer and which one doesn't sound very nice. And no pressure, but if you get this wrong, then my entire investigation is pointless. Okay, the first one. Oh. And the second one. Okay, who thinks the first one sounded better? Who thinks the second one sounded better? Okay, phew. What I just played to you is a dissonant and a consonant interval. But what makes them so dissonant or consonant? What makes them sound good or bad? Well, I wanted to find out how we could rank them mathematically instead of this subjective ranking. And after calculating and researching, I found that it is based on frequency ratios. More specifically, the complexity or simplicity of the frequency ratios. Two examples of this are a constant interval, the perfect fifth, has a ratio of 2 to 3, a simple frequency ratio, and a dissonant interval has a ratio of 32 to 45. This is a complex ratio, and yes. But what triggers us to react to frequency ratios, and how does our brain react to frequency ratios? What parts of our brain are stimulated so that we can provoke an emotional response? Well, this is all to do with the limbic system. The limbic system is the part of our brain which deals with emotional and behavioral responses. And more specifically, two specific parts of the brain are stimulated during these two intervals. And I know that in every single lecture, everyone always says that you can't assign a certain function to a certain type of the brain. However, I am going to do so. So the nucleus accumbens is stimulated when listening to consonants. Then it secretes dopamine, and therefore we have a pleasurable reaction to it. The amygdala, however, is stimulated when we listen to dissonance, and therefore creates sensations of fear or displeasure and unsettlement. But I still had this question of why do we react to frequency ratios? Why does our brain care about the simplicity or the complexity of frequency ratios? And what is the link between these two? And I created my own hypothesis after looking at other sensory types, such as color or aroma, which can also provoke an emotional response in the limbic system. For example, red can stimulate the amygdala and also create this sensation of fear. And so I thought that perhaps this was an evolutionary development which we had evolved to have, comparable to warning calls in nature. And yes. So, what have we learned today? Well, first of all, we found that we can make a mathematical ranking of frequency ratios from most simple to most complex based on the consonance and dissonance. We found that it is to do with the limbic system. This is what provokes an emotional response. And finally, we found that this could perhaps be evolutionary, meaning that we may have developed this in in order to perceive danger, for example, when hearing distance. Okay, thank you very much for listening.
It is always good to learn more about our Jonk Fuerscher. This is why please welcome back Arvids Andersens for the usual informal exchange.
Arvid, hello and thank you very much for the presentation earlier. We have some questions to ask, so basically it would be interesting to hear what was the starting point for your studies on toothpaste?
Yeah, so I mentioned in the presentation that I'm interested in dentistry as a prospective career. Next year I'm going into dental school, and then afterwards, of course, I'll be working as a dentist. So I thought, well, I should already pursue this passion. I should look at things I can already look at, toothpaste and etc., because I use it every day. So I was interested to see the chemicals inside of it and how, of course, it works to inhibit bacteria.
Is there a specific type of toothpaste that you found in your research that you would recommend?
Yeah, so what I tell people when they ask me this is just any toothpaste with stannous fluoride, to me, is good, because I did another lab where I was investigating toothpaste, putting it in different sodas and just seeing how the enamel erodes. And through that, I was doing a lot of research online, and I found that one common denominator is that stannous fluoride is really good at protecting the enamel. And of course, even when you have bacteria, their byproduct is what causes the issue, acid. And stannous fluoride will protect it. So when you look at toothpaste, look at the back and look for stannous fluoride. It's similar in all languages, stannous fluoride. So that's my recommendation.
Advice. Very good advice to give forward to our audience then. Was there any obstacles occurring in your studies or any surprises you didn't expect to come up?
Well, I think I didn't expect that much contamination. I thought my aseptic technique was pretty good, but in my data table, I don't know if you noticed this, it was pretty small in the presentation, but some of the Petri dishes had zero growth of the bacteria I was expecting. When I looked at it, it had a lot of oddly shaped bacteria that I just disregarded, and of course that's why I had zero. So I didn't expect that much contamination. So.
Are you going to participate again and further your project?
So, well, this depends if I have the time. So dental school is very busy. A lot of people know this. And of course, very demanding with the education. So if I have time to go into a lab, to create a new method and really deepen my research, then of course I'll participate next year. And I really hope I can. But this is kind of out of my control. But I really hope I can, because this event is wonderful. I think the way it's set up, it's very professional, and it's a great opportunity. So I hope I can.
That's great to hear, and wishing you all the best for your future career, for your studies then, and thank you very much for coming here today and just participating.
Yeah, thank you guys for organizing.
Ladies and gentlemen, thank you so much for following the 51st National Jonk Fuerscher Contest. It's now time for a short musical interlude by a very talented group of violinists, Anna Molitor, Catherine Posing, and Jo Schmid. But did you know that Anna and Catherine, together with Elmina Kochan, are also our former Jonk Fuerscher? Let's invite them on stage for a small interview.
Hey. So, also ich mache ein Interview auf Lützeburg, damit fühle ich mich einfach. Erzähl mal, wie geht es dir?
Es geht ganz gut. Wir sind immens froh, dass wir heute sehen und von den neuen Projekten zu hören, die neuen Jonk Fuerscher kennenzulernen.
Ja, das freut mich. Aber letztes Jahr stand dir halb der Bühne auch mit deinem Projekt. Könnt ihr euch noch daran erinnern, was für ein Titel es war, also um was es sich da gehandelt hat?
Also das Projekt hat gering bauen geheischt und es ging halt darum, für ein bisschen mehr nachhaltig zu bauen, wie mit Beton. Und auch zu isolieren. Und das ist halt eine Pflanze, die heißt Miscanthus. Aber wenn ihr die mit Kalk und Wasser mischt, dann kriegt ihr einen Zug Beton, mit dem ihr halt bauen und isolieren kann.
Wow, das ist ja wirklich interessant. Aber was für einen Preis habt ihr dann für den Projekt gekriegt? Erinnert ihr euch noch?
Ja, wir hatten eher auf COP26 dürfen zu gehen.
Aus Schottland, also Glasgow.
Oh, cool. Glasgow ist wirklich schön. Und erinnert ihr euch noch an die Experience? Also, wie war es? So ganz kurz.
Es war einfach eine immens flotte Experience. Wir durften ganz viele neue Leute kennenlernen, mit ganz vielen Leuten schwätzen und von verschiedenen Projekten hören.
Und auch ganz viele verschiedene Länder gesehen, was die alles zum Klima beitragen, was wir alles nicht wussten. Also wir haben viel gelernt draußen.
Ja, das ist ja die Hauptsache bei dem Ganzen. Aber ja, dann noch ganz leicht froh. Was gebt ihr dann Jonk Fuerscher, also junge Leute empfehlen, die bei dem Heiter-Konkurs mitmachen wollen?
Ich gebe so einfach das machen, was den gerne möchte und einfach sich trauen und ja.
Einfach machen und auch wenn es schief geht, das ist nicht schlimm. Es kann ihn immer alles verbessern. Das ist ganz normal an den Scienzen und den Naturwissenschaften, dass Sachen schief gehen. Und einfach weitermachen, dabei bleiben. Und, ja.
Das machen wir ja heute. Ja.
Absolut. Ich sage euch vielmals merci für den schönen Interview. Und dann gehe ich weiter und Marie.
Thank you so much for that. Now, we welcome on stage Viva Lux Musica for their incredible debut performance as a violin trio. Thank you so much to Viva Lux Musica for this amazing performance. And we are so lucky to have our former Jonk Fuerscher back with us for this edition of the contest. Now, let's come back to the science contest. Benjamin Frank from the Lycée Classique de Diekirch will present his project to the members of a jury. It is called What are Virophages? Let's find out here shortly.
Hello, my name's Frank Benjamin, and I'm fascinated by small things, so that's why I'm presenting virophages. I'm trying to keep it simple because I explained it to my mom like 5 times already. I'm still not sure if she understands. So, how do they work? So first, I'll introduce the main characters. First of all, we have this tiny little cute being that's called a virophage. Then we have its enemy. It's the giant virus, which is basically just a virus on steroids. Followed by the host of the giant virus, it's this lovely little amoeba. And once infected with giant viruses, there's 2 outcomes. First, it will survive, grow stronger, and become a surviving amoeba, or it will explode. And then you have the 4th player, and that's the surrounding, and it's filled with other innocent little amoeba. So, this is how the infection all starts. The giant virus is, like I said, pretty big, and that's why it has another mechanism to enter a cell, like any other virus would. It's basically as if it's eaten by the cell. So it looks like a tasty snack, but it isn't. And once inside, it will create a viral factory where it will mass produce itself, and the result, kaboom. And after that, you'll have the surrounding amoeba surrounded by numerous giant viruses, and this will all just repeat until everything's done. So now, let's add into that equation a virophage. A small little thing, almost particle-like once it was discovered, that will latch onto the giant virus. And upon entering, you'll see the difference. The viral factory not only produces giant viruses, but also virophages. And virophages are produced more easily and faster, and the resources are therefore much more quickly empty, and therefore there will be a diminishing number of giant viruses produced, sometimes even up to 70%. And you'll have a strong surviving amoeba that will release a few giant viruses and a lot of virophages that will once again latch onto the giant viruses, and so forth. So, big deal. But that's not all. The important part, which is really interesting, is how they all play together in the ways of DNA. Because here you see that the virophage will implement a sequence of its DNA into the host amoeba, and once it survives, it will grow stronger, obviously, but also have a defense mechanism against those giant viruses. But not everything's so good, because the same can happen to the giant viruses. They will adapt. They will use the virophage to grow resistant. And in general, there's a lot of splicing and changing of DNA that's happening. And therefore, they may even be the cause of diversity in microorganisms and therefore bigger things in general. But it's a really recent discovered thing, and so it's been, the giant viruses were discovered in 2008, so it's not long ago. And there's still really a lot to be understood about them, a lot to be researched. We don't even know how evolution plays a role in the creation of these little things. But it's just a really, really interesting topic, and I really hope that I'll get to figure out some more stuff, and maybe we'll see each other again. Thank you.
You remember Daniel Jankin from before our musical intervention? Well, let's have a small chat with him and find out more about his project.
Hello, good morning. Thank you for the earlier presentation. We'd like to ask you a few questions. So, what was your inspiration when combining the idea of mathematics and music?
Well, I've done music for a while now, and I just started this course on counterpoint. And it heavily involves these concepts of constants and distance. It's basically composition. But I never really understood why they actually exist. And I wanted to know, because it seems quite random, you know, which intervals were constant and which intervals were distant. And I wanted to find out, you know, how it worked.
Do you play any instruments or practice music as a hobby?
I played viola and piano for a number of years, but now I'm doing more theoretical music.
And looking into your personal interest, are you more interested in science or art, or both, then, the combination of both?
I'm very interested in, you know, kind of psychology and how different like sensory, like music or other senses have an impact on our emotions. But I'd say I'm more interested in science.
Right. And then looking ahead, following your participation here, looking ahead into your future career, would your presentation have any relation to your future plans, your studies you might look into?
It's got me interested in neurology, definitely. I want to do the basic thing of like engineering or something. Well, not basic, but yeah. But no, I found the limbic system especially to be really fascinating, yeah.
So do you know what you're going to study or continue afterwards?
Yeah, well, it's kind of either engineering or neurology right now. That's what I'm studying, yeah.
Did you face any difficulties during your project?
Yeah, the main difficulty was because I had an experiment I wanted to do, where I'd find for myself which parts of the brain were, you know, activated when hearing certain intervals, but I wasn't able to do it because it requires quite a lot of expertise, which I don't have yet. Yeah.
Now, after your experience here in competition, is there any advice you can give to future participants?
Definitely choose something which you're interested in, because it's a lot of time, and if you're not interested in, I don't know, it doesn't really take off very well. But I mean, yeah, just have fun.
So do what you love, and then this is what you're good at.
Speaking about that, how much time did you spend on your project?
Oof, the mathematics part took a long time. It took me so long to find some type of link between the frequency ratio stuff in the brain, because I really had no clue, you know, why it would work. So it took a lot of reading through, you know, different articles, which were not really at my level, to you know, get some type of idea.
Do you plan to continue on your project and maybe participate next year in the contest?
I might try something a bit different, perhaps related to music and emotion in some other way, or maybe something completely different. Or I might maybe continue this and try and find you know, evidence for the hypothesis I made at the end.
Brilliant. Yeah, thank you very much then for taking the time for answering our questions. That is surely of interest to our audience.
Congratulations for your project, and then good luck for tomorrow.
Thank you, thank you.
Thank you very much.
Up next, we have Federica Maestri from the University of Luxembourg, who is now going to present her project titled ALS, When Genetics Epidemiology Comes Together. She seeks to answer the following question: to what extent do mutations and socioeconomic status correlate with the onset of Lou Gehrig's disease?
Good morning. My name is Federica, and today I would like to walk you through my research about amyotrophic lateral sclerosis and its possible causations. ALS belongs to a large group of neurological conditions known as motor neuron diseases, which result in the gradual deterioration and death of motor neurons in the brain, brainstem, and spinal cord. Motor neurons are essential communication links between the brain and voluntary muscle movement. Consequently, people affected by ALS have difficulties in breathing, swallowing, chewing. They burn calories at a faster rate. But they retain their ability to perform higher mental processes, such as reasoning, understanding, remembering. And this is extremely significant because it shows that even though they lose physical control, they are completely aware of their degrading conditions. Scientists have not yet understood why ALS strikes just certain people and not others. So, when it comes to rare diseases, in Luxembourg, there are about 5,000 to 8,000 rare disease cases that have been identified. And a key event at national level was the European Conference on Rare Diseases within the framework of the European Presidency of the Grand Duchy of Luxembourg. To go back to ALS, age, gender, and ethnicity all represent risk factors. Men are more likely to get diagnosed with it, even though this difference disappears with age. Uniform rates can be found in Caucasian populations in respect to the other ones, and 90% of the cases are considered as sporadic, which means that the disease occurs randomly with no clear correlation to the risk factors and family history. The remaining 10% inherits it through a mutated gene. So as for now, hyperlemia, smoking, farming as an occupation, strenuous physical activity, epidemiological modifications, and exposure to environmental factors have all been identified as possible risk factors. So the first causations that I've investigated was the mutation of the TDP43 gene, which is an heterogeneous nuclear ribonucleoprotein. So this gene is involved in neural genome maintenance, in the repair of DNA double-strand breaks, and its toxicity helps regulating the levels of RNA binding proteins implicated in RNA splicing. So what happens is that the aberrant systolic sequestration of this gene leads to protein aggregation, and protein aggregation leads to genomic instability, which results in neuronal death. The second cause that I've investigated was the mutation of another gene, the superoxide dismutase. And it has been shown that it's the gene responsible for producing the mutant superoxide dismutase protein that's responsible for, that might become toxic. And this gene is not only linked to ALS, but it's also linked to other neurodegenerative diseases, such as, for example, frontotemporal dementia. And this study was conducted on mice, and it has been shown that even when the quantity of mutant gene was less than the quantity of non-mutant gene, the disease was still developed. And therefore, this kind of mice might help us understanding the pathogenic pathway of the disease. However, we have to point out, to mention the ethical part that comes with working in mice in science, and the fact that there are some studies that have shown and are showing that mice might not be such a good model for neurological diseases as we thought. But for example, zebrafish could be. The last causation that I've investigated was the epidemiological trend. So in a paper published by the US National Library, it has been shown that there will be a 69% increase in cases in the next 25 years. So the question that arises is ALS a disease of aging? And the answer according to this paper is yes. But according to other papers, the answer is no, because yes, there will be an increase in incidence, but this is not due to the fact that the ALS is a disease of aging, but the fact that ALS affects a vulnerable group within the population rather than being a disease of aging. So for sure, survival rates will most likely go up due to improvements in healthcare. And another thing that we have to consider when analyzing data is the fact that there might be some discrepancies in the methodologies that have been used by the different countries to gather their data. So to conclude, there's a huge gap in literature. For example, the mutation of the 2 genes was only investigated in cases of sporadic ALS, which represents only 10% of all cases. And even though I found that socioeconomic factors and geographical prevalence might have a connection, not enough data is still available to understand which factor is more relevant. And genetics and environmental factors are seen to be playing a role, but ALS does not have any racial or ethnic boundaries, which points out the controversy of studying this disease. And therefore, large-scale studies are needed in order to allocate healthcare resources more efficiently. And therefore, the next step in scientific research would be the assessment of the correlation between a lineage, environment, and ALS phenotype. So, as for now, artificial intelligence, we might say that artificial intelligence might speed up the research on neurodegenerative diseases. But the direction in which AI and the analyzing of data is going is we want to eliminate biases. And by bias, we mean bias related to gender, ethnicity, and socioeconomic factors, for example. But the thing is that when it comes to neurodegenerative diseases, where we have these factors whose relevance has not been fully evaluated yet, we cannot disregard this data in order to prevent biases, because arguably, they might hold the key for a cure. Thank you.
Participants on stage, let's go back to Benjamin Frank, who will tell us a bit more about his scientific interests.
Okay. 1, 2, 3. Happy birthday to you. Happy birthday. You did not expect that, did you?
No, not at all. Thank you.
How was it to present actually during the birthday? Like, you should be partying with friends right now. What are you doing here?
Well, I think I got some priorities, and yeah, I've been really looking forward to today to present, and yeah, so partying can come later.
Okay, right. So it's kind of a birthday present to come here for you as well, right?
Yeah.
Okay, so we thought about like buying you a pie or something like that for the science. But also, thank you for participating. You know, like the viewers can actually see an interview of you on internet that came out. How did you feel about that?
It was, I was a bit nervous, to be honest, about being released and everything, but I got really a lot of great feedback, and I'm really happy about it.
Yeah, I invite everyone to watch it because it's a good insight. And a last point about like your performance or for preparation is like you actually participated, as we can see in a reportage, to the Science Next experience that we organized at the foundation. Can you tell us a bit more about it?
First of all, it's just great. All these workshops that were set up, they really are educative about things that wouldn't even really come to my mind, because like getting the ideas to even start a project, that was one of the first workshops, and it was just amazing. There was great performance in it, and yeah, I learned a lot. And all of these things that I kind of got through all these workshops were really useful to me, not only in this project, but also in general. Yeah, with school and in my private life, like there was a workshop about presenting, how to present properly, which is also really useful if you have to present in school. And yeah, so...
I had a chance to watch a tiny bit of those presentations and workshops, and actually, you know, like when you see them like online, then you think, okay, maybe I don't need that and stuff, but like when you're there, you realized how much there is to learn about the topics. And yeah. Do you have some questions?
Yeah, first of all, thank you for the great presentation. I think it was very impressive how you brought across that complex topic in such a, you know, vivid and fresh way. Obviously, for me, it was the first time hearing about that topic. But did you do the graphics by yourself?
I did. I was struggling a lot with how I would do and structure my presentation. I was sitting there and I was thinking to myself, it's a complex topic, but I really don't want to get into all those details about like the DNA transfers and everything, because that's actually a really huge part. So I asked myself, okay, well, what can I do? Like, just try to, because only 5 minutes, that's not a lot. So I thought to myself, okay, I'll just explain how they work. And since it's not really that researched yet. There's not a lot of cartoony drawings on the internet. So I had to just sit down with a pen and paper and get at it. And yeah.
I heard like recently on radio podcasts and things like this that people have more and more interest in virophages, but do you think the pandemic, like the last 2 years, had an impact on your choice for the topic, or?
Definitely. I watch a lot of YouTube, and there was a video from Kurzgesagt, which I can only recommend, about bacteriophages. And I was looking at it, and I was really impressed, and I just thought to myself, well, works really well. So we're in this pandemic, maybe we could speed something up if we had something similar, just that it works with viruses. And a friend of mine just looked at me and said, well, I heard there's virophages. I don't know nothing about them. And I looked into it, and it was just really, well, I was astonished by all the complexity to it, and also the potential in it. So yeah.
Would that be something you would like to pursue for a next issue of the Jonk contest?
I'll try to now get into the practical ways of the research, and yeah, therefore also then maybe preparing another funny presentation for next year.
Back.
Yeah, I'm coming back.
We are glad.
Sounds like a plan. Looking into that topic, is that something you consider also for your future career? Look into further studies? Or?
Maybe it's really fascinating. I myself am more of a marine biology guy, but all these tiny things, I mean, virophages and giant viruses also rely on aquatic environments, so maybe there's some overlapping. Yeah.
Okay, then thanks a lot for your presence, for your presentation, and we wish you good luck for the contest.
Thank you very much.
Thank you so much.
Now, Sarah Compagnivala and Elena Lugiens-Maché from the Lycée Michel Lucius are going to present their project titled Essential Oils, Reality or a Hoax. Since ancient times, essential oils are often used in aromatherapy, by inhalation, or by application to the skin. Essential oils were seen as the medicine of the future, as nature's healing powers, but have also been labeled as a hoax. But let's hear from the participants about their findings.
Essential oils, a reality or a hoax? Hello, I'm Sarah Compagnivala.
Hello, and I'm Elena Lugiens, and today we will be presenting our project about essential oils.
We performed a total of 2 experiments, one testing the effects of lavender essential oils on Daphnia magna, a water flea, and the other on humans.
So, what exactly is the problem we're trying to investigate? Many people do not believe in essential oils and see them as a scam, accrediting any effect of them to a simple placebo.
So, to see what they actually did, we performed a few researches, and all we could find were mixed conclusions. So, we did what we had to, and we performed a small investigation.
What exactly are essential oils? Essential oils are concentrated plant extract, which are made by distillation of a specific plant, which results in a liquid that has the same chemical properties as that plant. Essential oils have many benefits, and there are a great variety of essential oils. They have been used since ancient times, especially in old Oriental medicine.
Therapies that use essential oils to treat psychological disorders prove their effectiveness. But like other clinical trials, a placebo must be taken into account. There are many different types of lavender. For example, as you can see here, in our experiments, we used Lavandula angustifolia, which is used to treat anxiety, stress, and insomnia, whereas other studies use Lavandula latifolia, which treats pain and inflammations.
For our first experiment, we investigated the effect of lavender essential oil on the heart rate of Daphnia magna. Daphnia magna are small planktonic crustaceans that are commonly called water fleas. Their average heart rate is 200 beats per minute, and they're ideal for heart rate experiments due to their invisible body. One can perfectly see their heart and their beats.
This is what it looks like under the microscope. And here is its heart, as you can see, very visible. Our hypothesis for this experiment was that lavender essential oils would have an effect on the heart rate, specifically decreasing it.
As you can see here, there's a significant decrease once you add the lavender, supported by a statistical t-test.
Since this supported our hypothesis, we decided to carry out a second experiment, testing the effects of lavender essential oils on humans.
So, the human heart rate is affected by many factors. One of these factors can be a placebo. Our subjects were all females between the ages of 15 to 18.
We had 10 volunteers divided into 2 sessions with 5 people in each. Both of these sessions were performed on different days.
To test that effect on the effect of lavender essential oil on our subjects, we set up 3 rooms. In these 3 rooms, in the first room, we had no presence of lavender, and our subjects did not know what we were investigating. In the second room, we diffused the lavender but did not tell our subjects about it. In the third room, the presence of lavender was still there. The oil was still diffused, but we did tell our subjects about it and clear them up about the benefits of lavender essential oil.
As for the method, upon entering the room, the volunteers were wrapped up in Pascal heart rate monitors, as shown here. We recorded their heart rate for 5 minutes and then asked them a few questions. How are you feeling? What were some thoughts you had in the past 5 minutes? And do you feel tired? Then we took a short break and moved on to room 2. In room 2 and room 3, we did the same exact method while a break in the middle as well. Then we compiled our data and calculated the averages. There is a pattern seen, but the differences in heart rate between the rooms can be argued to be negligible. But as you can see, there is some sort of pattern. There is a decreasing heart rate from room 1, where lavender was not present, to room 2, where lavender is present, but the volunteers did not know about it, to room 3, where lavender was present and they knew about it.
40% of our subjects reported to be really stressed at the beginning of our investigation. In room 2, of these 40%, 75% reported to feel less stressed and more calm. In the last room, in room 3, with the presence of the lavender and the information about it, 90% of all of our subjects felt much more relaxed and much calmer.
But when we look at individual data sets, we see different patterns. Most of our data looks like this, like subject 1's, where it supports our hypothesis because the heart rate decreases from room 1 to room 2 to room 3. Specifically here, subject 1 said they felt stressed and anxious in room 1, but felt more calm and more relaxed in room 2 and room 3.
Very few of our data also look like this graph. Here you can see that subject 8, the overall trend of its heart rate is quite constant. Subject 8 also reported not to have had any change in mood or any change in stress levels. The lavender does not seem to show any effect on their body.
But not everything went well, so what went wrong?
Short exposure. Before we started recording our subjects' heart rate, they were only exposed to the lavender for about 1 minute, which might have not been enough time for the lavender to show any effect on their body.
Secondly, some volunteers said they could smell something in room 2. If they knew what lavender smelled like, or if they knew the benefits of lavender, it could have affected the heart rate.
Varying resting heart rate. So every individual has a different heart rate, and when you consider the results of all of our subjects together, it's quite difficult to compare them. To improve this, a percentage rate could be calculated.
To completely refute the lavender as a placebo control should have been carried out. This is what it would have looked like. We would have had room 4, where they would have been told lavender is present, but actually no lavender would have been present. We would have carried out the same method as we did in room 1 and 2 and 3 and compared the data. If there was a decrease in heart rate compared to room 1, where lavender was not present either, then lavender could be concluded solely as a placebo.
So, what is the future for essential oils? Essential oils could be used as alternative medicine for small ailments and taking advantage of the wide variety of essential oils. Essential oils are also helpful during medical procedures. We have found many studies that conducted research to see if lavender essential oil reduces the anxiety of a patient before a medical procedure, and many of them had really positive results. In general, essential oils should be taken seriously. They are a reality. They are not a hoax. And if we start having that defining research, we can take the full potential of essential oils. Essential oils could be used in schools and exam halls to reduce anxiety, or in hospital waiting rooms.
All in all, essential oils, specifically lavender, has an effect on heart rate, specifically decreasing it, as shown as Daphnia magna and humans. And even if there is a placebo to be taken into account, it does not mean it's a bad thing. Placebos have an effect on almost all symptoms known to mankind. They cannot be given an overdose and do not have any serious side effects. So, in short, placebos hold the prize for the world's most protein, safe, effective, and cheap drugs. So, if they can enhance the treatment, then why not? We acknowledge that essential oils cannot replace modern treatments, but they could be used to enhance them, for example, in chemotherapy or to reduce post- and perioperative anxiety. Thank you for listening.
Now, let's have a quick chat with Federica Maestri to get to know her a little bit better.
Hello, Federica. How are you?
I'm good, thank you.
Thank you very much for your earlier talk. We're happy to have you around and to ask a few more questions. So, what I'm curious about is to know, what was the starting point for your research on ALS? What was your motivation?
I had a family friend that was diagnosed with ALS, and therefore I was curious about investigating more about this disease and the fact that there's no cure for it. And this is what made me start my investigation. And when I realized that there's actually a connection between the way we analyze neurological diseases and artificial intelligence, the way we analyze data sets and data, made me want to continue with my research.
Have you faced any specific difficulties during your project?
Yes, I feel like as in every neurological disorder, it's really hard to find the right papers, and also since the literature is a bit limited, it was hard for me to find many studies and compare the studies. Some studies are controversial. Some say one thing, some others say another thing. But I think it's not only because of studying ALS, but in rare neurological conditions in general.
And the fact that there's so little literature, is that a motivation for you to also continue your research after the project? Would that be something to look
Yes,
into?
I feel like also, the more people that start studying about ALS, the more the scientific research will be motivated to continue with experiments and studies about it. And I feel like there's always has to be a starting point somewhere. So yeah, I definitely believe that having people who start researching more about these conditions is definitely an improvement and a step forward.
It might not be completely related to your project, but during your research, have you discovered things that you should pay attention to in terms of persons that have this kind of disease?
What do you mean?
Like, for example, preconceptions people have, or things that people handle a certain way and that you should rather not do?
Yes, I believe that the word rare disease make it seem like something really rare, and therefore people never think about the fact that there might be a starting point of these diseases, and therefore the first symptoms are sometimes disregarded because they're not considered important. And I feel like maybe a bit more awareness about the fact that yes, these diseases are there because are rare because there's a small part of the population that's affected by them, but the fact that we shouldn't completely disregard them and maybe think, this cannot happen to me. Yeah, I feel like it's a bit a misconception about rare disease and the fact that there are not many people that are affected by it, and therefore it's not that important.
So to be more aware about it.
Mm-hmm.
Also then talking about medicines and help there, so that I understand that it needs some communication to be done and some advice in early stages so that people become more aware, and also practitioners become probably more aware.
Yes, definitely artificial intelligence that has already been used for neurological conditions, because, for example, digital microscopes might help identifying dying neurons before humans can. Therefore, there are some steps that have been done forward, but of course, there's much that has to still be done. Yeah.
Do you know if you're going to continue on this project and maybe participate next year?
Yes, but I don't think right now I'm studying computer science at university, so I don't have that much. I don't have that much possibility to continue with this research from a biological point of view. But definitely the analysis of data sets when it comes to medicine might be something that I would like to continue investigating.
Brilliant. Yeah, then we thank you very much for being here with us. Thank you. We wish you all the best of luck for tomorrow then.
Thanks.
Congratulations for your presentation also.
Thank you.
Our next participant, Petra Strager, from the Lycée Michel Lucius. Her project is titled Bacteria with Headphones and investigates the effects of sound in form of music on the growth of E. coli. Welcome now, Petra.
Hello, everyone, and welcome. Before I actually start with my presentation, I would like to ask the audience to please raise your hands if you enjoy listening to music. Can you please raise your hands if you play or have played an instrument? Thank you. I myself, like many of you, enjoy music, and I have played the cello for the past 8 years. So I strongly believe that music affects all of us. And there has been a study on Mozart's music, how it affects people, and it has been shown that it actually improves their well-being and it makes people more intelligent. Sorry. It's not working. Thank you. But not only does it have an effect on humans, it also has effect on animals. In 2007 in Spain, a study has been conducted and cows were exposed to Mozart's music. And essentially what happened was that cows that were exposed to music produced 30 to 35 liters of milk per day, compared to cows that weren't exposed to music, and they only produced 28 liters of milk per day. Now the question is, what happens when we put this in context with microbes, with bacteria? And this is what I will present to you today. My project is investigating essentially how is bacterial growth affected by music. So first with the hypotheses. My hypothesis was that music will indeed have an effect on bacteria. And according to other scientific articles that have been done before, classical music would have had the greatest effect on bacteria. The IV in this case was the musical genres used. So I've used 4 musical genres. And the DV was actually the growth. So the growth of bacteria was measured. Some of the controls, for example, were the incubator, where bacteria grew. Thank you. We had the same sets of headphones for all of the bacteria, and they all listened to the music for the exact same amount, so about 8 hours for 4 days per week. So every week there was a different genre of music. I would like to spend a minute to talk about the bacteria I use and the music I use. So once again, I would like you to raise your hand if you enjoy jazz. I would like you to raise your hand if you enjoy classical music. I would like you to raise your hand if you enjoy EDM, techno. And I would like you lastly to raise your hand if you enjoy hip-hop. So these are the 4 genres that I have used in my experiment. And the reason I chose E. coli was because it's very easy to handle, it was easily available to us, and it's essentially the first choice for researchers when it comes to bacteria. The procedure itself was very simple. So first it was pouring the plates, which was basically melting the agar, pouring it into the plates, and then waiting it to cool off. After that, we had spreading the plates, and this was, now that you have your plates with the agar, you use a pipette, and in my case, it was the L-shaped cell spreader, and I used it to just spread the bacteria across the agar. And lastly, it was about bacteria listening to music itself. So all I did was downloaded music on the MP3, connected it to headphones, and put it over the bacteria plates for about 8 hours every day. So these are the results, and classical music showed to have the greatest result, as we can see on the left, compared to the control experiment, because we can see a lot more colonies in the classical condition. And this was followed by hip-hop. And hip-hop music also grew more colonies compared to the control condition. EDM and jazz conditions either had no significant difference, or there just was no difference at all compared to the control conditions. But now the real question is, why is all of this happening and how is it happening? So we, of course, know that bacteria don't have ears. They can't hear music like we can. But there are theories about them having these mechanisms or receptors that are able to respond to mechanical stress. So essentially different components in music like tempo, which is the speed of the music, the mood, the lyrics of the music, they all have an effect on the growth. And in more technical terms, we have music, of course, which is sound waves. And these sound waves are in nature mechanical stress. And what this mechanical stress does to bacteria is it enhances their metabolism and it enhances their permeability of the plasma membrane. When we have increased permeability of the plasma membrane, what happens is that it leads to signal transduction, it leads to substrate transportation across the membrane, and this is what essentially promotes cell growth, cell division, and lastly, metabolism. So even though there are many things that we still don't know about this, there are many things that need to be researched. One thing is for sure, music indeed does have an effect on bacteria. Thank you.
Before the lunch break, let's first hear back from our contestants Sarah Kompanivala and Elena Lucians-Maché. And then we will have Petra Straga tell us more about her project in scientific interests. Thank you.
Hello. We were going to ask a couple questions about your project and about you and how it went. First of all, how did you guys get acquainted with alternative medicine and specifically essential oils?
So my mom, we are 8 children, and she really likes essential oils. She used them a lot on like all of her daughters. For example, lavender, she would use that a lot when we had like insect bites, like the other one, the Latisfolia instead of the Angustifolia. When we wouldn't sleep and we would be annoying her a little, she would diffuse it around the house to calm us down and get to sleep better. But yeah, it's mostly my mom, and also she had some experience in the family with essential oils.
We also are in our last year, so we have a lot of anxiety, a lot of stress and all that, and we just wanted something that could help us, but like not the pharmaceutical medicines like chemicals. So I was like, your mom did that when you were younger, so let's try that out. Let's see if it actually works. So.
Sounds good.
Did you see any surprises in your research? Any findings you weren't expecting before?
I genuinely didn't think the whole essential oils would be like measurable in any way, because I knew it when I was a child. When I grew up, I noticed that people didn't really take it seriously. So I kind of took that as like, oh, it was just because like placebo. It's my mom. She's taking care of me. So I didn't actually think it was like something I could measure, something we would see on our graphs. And I think that was kind of a surprise to especially me. I mean, she was sure we would see it. That's why she offered to do the whole experiment.
We had a little argument before, like, it's gonna work. No, it's not. It's gonna work. I'll prove you wrong.
and it did end up working out well.
So overall, the cooperation went fine with you two?
Yes, we work perfectly together. I don't think we ever had like a problem.
No, I think we compromise a lot.
Yes,
we compromise a lot.
Yeah.
But it always ends up working out perfectly.
Sounds good. Did you face specific difficulties during your project?
Yes, the Daphne magna, they kept on dying. We had to decrease the concentration of the oil by so much. But I think in the end we found one that worked, and they stayed alive until pretty much the end. So.
And then getting human subjects, which are students, which have a tight time schedule, getting them to work with us for an hour before their lunch break, during their lunch break, during their 15-minute, like during their breaks that they have, getting them to cooperate with us was also quite.
We had to bribe them at the end with chocolates. They're our friends, so they had to,
still.
Manipulation.
But yeah, that's why we only had 10 subjects, because it was really hard convincing other people to participate in something that they do not know what is going to happen. Like people usually don't like that.
Did your study motivate you to look further into that topic? You mentioned you've focused very much on lavender oil. Are you looking into deepening that research further, or was it also an inspiration for you for your further studies?
I think we're definitely going to look into placebo a bit more and how that works and why it happens and stuff like that. We'll definitely want to try out other lavender oils, but other types of alternative medicine, so pills maybe, and like other, maybe in your food, some spices, because turmeric helps with a lot of things, cinnamon helps with a lot of things, so definitely go into more of that side as well.
So not only inhalation by diffusion or anything, but maybe like oral ingestion, or other pathways.
Application as well and stuff like that.
Because there are so many things one could do. We really focus on that one to like get those control variables done to make it a reliable experiment. But there are really so many things one could investigate.
And you're in your last year of high school now. Do you still plan to continue the project and maybe participate next year?
We would love to. I would really like to do it again. I had a really pleasant time on it, although because of exams that are coming up, we did have a tight schedule, but I would love to do it again when I'm in uni. So.
I would love to do it too. We'll see how it works out for us both in uni.
Maybe one last question. I think we can see like an increasing demand for alternative medicine in society these days. Why do you think is that?
I feel like they're looking for something that is maybe not synthetic, something that's more natural, something maybe cheaper, and something they're more familiar with, you know.
They're often also looking for something that they can explain easier. Like if you look into nature, if you're trying to explain to someone, oh yeah, I did this in the like laboratory. But if you explain to them this plant, when you see this plant, it can do this. And if I take this plant and distill it and get that liquid, it will do the same. And I think often also alternative medicine promises not to have any side effects. Because if you look at like the most medicine we use, it has side effects, obviously. And then alternative medicine promises to not have any or very little side effects. So I think that's like one of the major things why they look to alternative medicine.
Yeah, so I think quite a relevant topic you have started looking into. Thank you very much for taking the time and speaking with us and answering our questions. I'm sure that was interesting for our audience to listen to.
Thank you very much, and then good luck tomorrow.
Thank you very much for taking interest in our project as well.
Hello, Petra. Thank you very much for your talk earlier. Yeah, first question coming up to my mind is really how did you get the inspiration, or how did you start with looking into your research topic initially?
Well, at first I had no inspiration for the project, but I really wanted to do something, and then I started looking up things online, and there was a similar project to this one. And yeah, I thought it was great because I like biology and I've been doing music for a lot of years, so I thought it was a good project for me. Yeah.
You mentioned you've been doing music. Do you play any instruments?
Yeah, I've been playing the cello for the past 8 years, but then along the way you also pick up some other things. So yeah.
Did you come up with any obstacles during your studies?
Yeah, actually, we kept. Well, I was working by myself, but my teacher helped me a lot. And during the experiment, there was a lot of obstacles, a lot of technical problems with the MP3s, and then the bacteria would get infected, and then yeah, that sort of thing. Yeah.
Yeah, I see. And yeah, when looking into that competition, despite the learnings from your research itself, can you give any recommendations to future participants which might be useful?
Yeah, I mean, I think you should really look into what you're interested, because if you work on something you don't like, then you're not going to get it done at the end of the day. So you should really do something that you enjoy and maybe something that you want to do in the future. Yeah. So that could help. Yeah.
Did you face specific difficulties while working on your project?
Well, other than the MP3s and the infestation, no. I mean, it was an easy project, but yeah, there were some obstacles like that along the way. Yeah.
Have you been working on it for a long time?
Yeah, I started in beginning of November, and I just finished before the February break. So yeah, a few months. Yeah.
And looking into your personal future, would you see any link to future studies now by your research? Biology, is that something you're looking ahead also for possible studies?
Yeah, I mean, I'm currently interested, I'm looking into the biomedicine direction, so the research direction. So I think this is why this competition is a great opportunity for me and for everyone else who's interested in science. Yeah.
Brilliant.
A bit closer in the future, do you know if you will expand on your project and maybe participate again next year?
Maybe, yeah. I mean, I thought it was really fun. It was a lot of work, but it was, yeah, really fun. So I hope I will, participate next year as well.
Perfect. And yeah, thank you very much for taking the time answering our questions, and best of luck for tomorrow also. And congratulations again.
Thank you very much. Thank you.
Ladies and gentlemen, welcome back to the 51st edition of a National Jonk Fuerscher contest. Now, let's welcome two of our next Jonk Fuerscher, Clarce Varmos and Alfiona Palacio, from the Lycée Michel Lucius. They will come on stage to present their project titled, Does the Moss Grow on the North Side of Trees? Stay tuned if you want to find out whether the direction of moss growth can help you to navigate.
Good morning, good afternoon, everybody. I'm Alfiona.
And I'm Clarce.
And we're talking about, for today, it's about moss, or in scientific terms, Bryophyta. Okay, it's the other way around. A-Y-N-T, okay. And next.
The main question of our project is, does moss always grow facing the north side of trees? Common knowledge says that moss grows to the north of side of trees. We decided to test this because
we always strive for adventure, and we are both adventurous and dauntless, except for public speaking sometimes. So one day while we were walking on a sunny day, we thought of what if we get too adventurous to an extent that we get lost in the forest without anything. That's why we questioned if
moss can help us navigate. We decided to test it ourselves to find out the most accurate answer. We started by
choosing 3 different locations in closed areas, open areas, and near pond areas.
And then the methodology. Most of the trees that we chose had moss only on half of the tree, and on the other half of the tree there wasn't any moss at all. So we chose to measure on the middle part of the moss. So first we would measure, we would stick our stick into the moss and put our finger near the edge of the moss, and then we would measure it using a ruler, and then we would
use a compass to determine which direction most of the moss grows. It's very essential, especially in this project. And next is
the 3 things we made sure to always measure. We would always measure the time, the date, the temperature, and the humidity.
The results shows that the thickest moss grows mostly on the west side, and the thinnest moss will mostly grow on the east side, following with northwest, southwest. And
then we also made this graph to show that in the different places which direction it showed. And for the exposed, all of them face the northwest, and for the pond, some of them face the southwest and some of them just face the west. And then for closed, it faced most directions, which is east, northeast, and northwest.
Looking to the results, you've seen that the results are affected by these factors. It includes the location, the humidity, vegetation, and temperature. Comparing to the 2 different areas, the closed areas and the open areas, we've seen that there's quite different results rather than the near pond areas. And we've thought that it may be because of the pond near it. And yeah.
And in conclusion, we learned that moss is not an accurate measurement of 100% of the time, but if you are lost, you could technically use it in exposed and a pond area.
Thank you.
Before we go to the next presentation, please welcome our second artist of the day, Oki, for a small interview. Let's take a minute to learn more about him, his inspiration and motivation in life.
pleasure to meet you, to have you here. Looking forward to see you perform on stage later. And yeah, we're here just to ask a few questions on what's upcoming. Maybe first of all, for you as an artist, I'm sure it's been a troublesome time now in the pandemics. How have you experienced that period, and what are your future plans now coming out of those exceptional times?
For me, the pandemic was, I would say, awful. It was really hard to really like, I think emotionally it was really hard for me. But I didn't let that like eat me up. So I tried my best to stay creative, to be fit, like physically the best I could be, because that was the only thing I could do. And I wrote a lot of songs. I still did work on projects, and I had like, coming out of the pandemic, I had a huge catalog that I could start releasing. So I would say emotionally it was awful, but I really managed to take advantage of it and use it to my advantage. Yeah.
Sounds good, sounds good. How did you actually first get into music?
Wow, that's a good question. I was born in Lagos, Nigeria, so I grew up with my grandmother. So we actually used to sing at church during church service, and I was still young, so I was like, I don't know, like 4 or 5 when I started singing. So at the age of going to the age of 11, I moved to Luxembourg, and I came here to meet my parents, and I joined the school choir, and right there they started like giving me solos and stuff. So it was a huge experience for me. It was like the best thing that could happen to me at the time. So I didn't know, so I met someone, and the guy, Jonathan Reichling, is a Luxembourger, and he actually gave me the opportunity to start producing music, start writing songs myself, and start, going through that creative process. And it led internationally, which is something at first I didn't have that in mind. I wasn't planning to start doing music professionally. So now I have a dream. I really, really want to break that. I want to break through here in Luxembourg and also internationally, because I've never seen really any artists do it. And with me, I can see that it's really progressing, and every day I'm closer to achieving that.
It's really good to hear.
Yeah.
So actually, I got a question. You said that you didn't plan to do music at first.
Yes.
So what did you have in mind as other possible career choices?
Yes, I had in mind to what everybody does, to go to university and to do social science, and I'm still doing that. Knowledge is important, and knowledge is the key. I understand that, but I think I've found myself, and I've found my passion in life, and I think that's just music. There's a lot I can do with music, like there's a lot I could surrounding music. I could turn it into business later, when everything blows up, I can turn it into something phenomenal here in Luxembourg. And I know it's gonna happen, because like I said, on Spotify, my streams are like I got support from Spotify, like the push, like performance-wise, I'm starting to like get there. I have my own band now, so I have a lot of possibilities. I'm working with producers outside of Luxembourg, and these are like huge people that knows what they're doing, and people that are already successful. So I think I'm just lucky, and there's still a lot of work that needs to be done, but I think I think I've just found myself, and I just want to keep going.
Great to hear, great to hear.
Promising, and a lot of energy deriving from your passion, which you can feel when talking to you. When looking ahead, can you give us any insights on future projects you're looking into?
Future projects, okay. Future projects, I would say this, there's a lot of performances coming, and it's gonna be amazing. Like, one I can give away already is I have this huge project with the Ettelbruck Primary School, and we're gonna like create a show which is gonna be a Nordstadt show. Like in the north, we have it's called Nordstadt. You know, you have Ettelbruck, Diekirch, like all the cities in the north, and all those kids will end up seeing our show, and it's also like a moment for me, for the kids to be introduced to my song, because that's like a sector or a demographic that I'm trying to reach to and get to connect with them more, because I think little kids, teenagers, they really love my songs, and they reach out and tell me, oh, yeah, it's really touching what you said there, or what we heard there, the lyrics is really touching. So that's one of the projects I can give away, but there are other surprises, and it's just amazing. So I don't want to say too much, but it's amazing. There are great surprises coming, and I think no one is going to expect this. I didn't expect it myself, so it just came out of nowhere, because I said to myself I was going to do it after my bachelor's, and I'm gonna take my time to like release an album, but I think no one is gonna expect this, so it's gonna be surprising. A lot of shows, a lot of commercial deals as well for me, and visibility.
So it's worth to stay connected and follow what you're doing.
Yes, please stay connected.
We will do that.
Yes.
Brilliant. Maybe last question. In this context here, having all those young talents around and young scientists being passionate about their project, is there any advice you can give them from your perspective as an artist?
Any advice I could give them? The advice I would give them is just don't give up. Like, even as an artist, from my point of view, I tried many things that did not work. I got many no's before I got a yes that opened many doors for me. So never give up. Always, like, I'm still in that process where I like finish a song and I'm like, oh yeah, this is a hit, you know, and I share it, and they'll be like, nah, it's not it, you know, like, and I'm like really disappointed. But you just have to keep going. You have to keep trying. During your, like, you know, scientists, they like try to like do a lot of tests, do a lot of runs, and you might get a negative result, and you might be frustrated, like, oh, I'm not getting the result I want to get. So don't give up, because who knows, you might be the next one to discover something that we don't know yet, you know. And don't give up, and try to open doors for other people. Think about the other people. That's what I would say.
It's really inspirational.
Indeed. Thank you very much for being here with us then, and all the best for your upcoming projects, and
thank you.
Stay tuned.
Yes, stay tuned.
Our next participants today is Alexandra Sten and Paulina Den Otter, coming from the European School. They investigated the sleeping patterns of high school students to see how that affected their health and academic performance. Welcome on stage Alexandra, who will now present their project.
Dear members of the jury, dear audience, my name is Alexandra and I am a student of the European School Lux 1. Together with my friend Paulina, we presented the project Analysis of Sleeping Patterns of High School Students and Its Effects. So why did we decide to do this? Well, this is why. Seeing as students at our school seem very tired, we decided to investigate further and make our own study on how widespread this problem was and what its causes and effects were. So to do this, we made our own survey on Microsoft Forms, consisting of 14 questions for students aged 16 or older at our school. To promote our survey, we made posters, which you can see here. We sent around emails, but we also asked students directly. So what were our results? Well, in total, out of the 512 students concerned, 146 responded to our survey. The majority of students who responded were female and aged 16 and 17. One of the questions we asked was at what time they woke up and at what time they went to sleep, both on school days and on weekends. From this data, we calculated that students on school days sleep on average 7 hours and 13 minutes. This is below the recommended amount of 8 to 10 hours for 13 to 18-year-olds. This means that students do not sleep enough. On weekends, however, we calculated that the average student sleeps 10 hours and 23 minutes. This means that students sleep longer on weekends, which makes their sleeping habits more irregular. The shift between the times they woke up and the times they went to sleep is also clearly seen on the graphs. We also asked whether students experience any difficulties falling asleep. In general, female students tend to have more difficulties falling asleep. One of the reasons for this could be the menstrual cycle, which causes the hormonal balance to change. We then asked students, in their opinion, why they had difficulties falling asleep. The majority of students responded that it was due to thoughts, stress, and concerns, which are psychological reasons. 42% of students admitted that they couldn't fall asleep because of irregular sleeping habits, which we had also observed on the slide before. Then we asked students what activities they did within an hour before falling asleep to find out whether there were some other reasons why they couldn't fall asleep. Here, we found that a majority of 60% used their device before falling asleep. 57% specified that they used social media, and 51% that they watched Netflix or other streaming services. All three of these activities expose the students to blue light, which is stimulating. Blue light disturbs the circadian rhythm, part of the body's biological clock, because it raises cortisol levels and it suppresses the melatonin excretion. At the end of our survey, we asked students for their opinions. This first student understands the importance of sleep, while the second student admits that their sleeping habits are irregular. The third student acknowledges that their lack of sleep has an effect on their school performance. So, what are the consequences of a lack of sleep? Well, firstly, as we have seen, students tend to wake up and go to sleep at different times during school days and during weekends. This means that their sleeping habits tend to become irregular. This could result in a vicious cycle, because as students try to recompensate their sleep on weekends, their sleep deprivation actually gets worse. Scientific research has also shown that a person's risk of getting depression or metabolic issues such as diabetes type 2 or obesity increases with their lack of sleep. Researchers have also proven that sleep is crucial for brain plasticity. This means that when students do not get enough sleep, they will have difficulties in processing what they learned during the day, including the material they learned at school. This may negatively affect their academic performance. So, what can students do about this? Well, students should not underestimate the amount of sleep they need, but instead they should get the recommended amount because it shows the best what they actually need. Sorry. Students should also establish a set sleep-wake cycle by setting themselves bed and wake times. And students should actually create a bedtime routine where they avoid any stimulating activities, like using their device. Above all, our survey shows that students do want to solve the problems with their sleep. More than half of students indicated it was somewhat too extremely important for them to change their current sleeping habits. Seeing as the school plays a central role in students' life, in the future, we want to show our results to the school, inform them about the lack of sleep of students and the problems with sleep students were having, and encourage them to take the well-being of students more important. But we also think that it is more important, we also think that it is important to continue our survey, our research with a larger age group and a more representative sample. Thank you for your attention.
Without further ado, let's have a little chat with Clartia Varmos and Alteona Palacio about the innovative ideas.
Welcome. So we want to know a bit more about your project, and as first question, I think I would ask that where did you get the inspiration and the motivation for your project?
It's based in our personalities. Especially her, she's very adventurous. And then just, as I said earlier, one morning we were walking and thinking about what we can do. And then there's like this tree, there's full of moss, and we thought, what if this would be the answer for it? So.
Sounds good.
And how did the teamwork go for your research? Did you have any specific roles in your team to follow, or how did that go generally?
It was good. Everything worked okay.
Yeah?
Yeah.
And when working together on that project, did you discover any surprises or unexpected findings?
Yeah, there were some unexpected things that we didn't really think about.
Yeah. Can you just tell us a bit more about what happened? What was that about?
What do you mean?
Like
examples of surprising findings?
Well, we found that we were surprised that in enclosed places that there wasn't as much moss like facing a specific direction. We thought that there might be more moss there because it's more dense and more humidity. And
yeah.
So that was surprising.
And we've learned that there's a lot of types of mosses, like thousands. And it's just not one type.
Mm-hmm. And have you looked into whether certain types of mosses look into a specific direction and others in a different one?
We haven't been there yet.
Could be something to extend your project, maybe. Do you plan to continue working on your project and maybe participate in coming years again?
Hopefully, yes.
The base of your research and the subject, is that something you would like to proceed further, maybe for future studies or future career?
Well, I definitely would. I really like biology and nature, and I think I might want to become a biologist when I'm older.
What about yourself?
I don't know.
Not yet. Yeah, I think you still have a few years to go also, but yeah, good to hear. Is there anything you would like to transmit as an advice to other students following their projects? Anything you can tell them?
Don't get lost in the woods. Always bring a compass so you don't have to use natural navigation. But hopefully in the future we could find out something more that we could use.
Brilliant. Good. Yeah, then thank you very much for giving us some further insights into your work and your future plans. And we wish you all the best for tomorrow. Thank you for being with us today.
And congratulations for your project so far.
Okay, thank you.
Our next participant's name is Isha Gönka from the International School of Luxembourg, who will be presenting her project called Optimizing UV Exposure to Inhibit E. coli Growth. In her project, Isha investigates the effect of UV light on E. coli growth by changing the time and intensity of light exposure on the bacterial cultures.
Good afternoon. My name is Isha Goenka, and my project is optimizing UV exposure to inhibit E. coli growth. I would like to begin by asking you a question of whether you would drink the tap water in Luxembourg. Many of our answers would be yes, as we feel comfortable drinking the water as we believe it's been industrially treated and it's safe to drink. This, however, is not the case for many people around the world, over 2 billion of whom do not have access to safe drinking water. A study found that 59% of water samples taken from Bangladesh contained traces of pathogenic E. coli. E. coli is a gram-negative bacteria which secretes Shiga toxins, which may cause neurological complications and acute liver failure. As such, there are over 2,000 hospitalizations due to E. coli infections in the US alone. As characteristic of gram-negative bacteria, the E. coli lipid membrane acts as a barrier to permeation of antibiotics, making it increasingly difficult to treat. While there are many water treatment solutions ranging from chlorination, filtration, and using active carbon, each one with their own advantages and disadvantages, the one of special interest to me was using ultraviolet light. This led me to developing the research question of how does UV exposure time and light intensity affect the growth of E. coli measured by the number of counted colonies after 24 hours of incubation. UV light is divided into 3 categories: UVA, UVB, and UVC, each with its own wavelength range. UVA damages DNA via indirect UV-induced damage, where reactive oxygen species are formed in a photodynamic reaction within cells, which can react with protein and lipid membranes within the bacterium, altering its permeability and eventually leading to apoptosis or cell death. It also generates adenine and guanine base modifications, changing the genetic code of the DNA strand and possibly leading to mutations or inactivation of the bacterium itself. Direct UV damage brought on by UVB and UVC causes DNA lesions formed by crosslinks between 2 adjacent bases of thymine and cytosine, covalently bonding these 2 bases, forming a dimer. If these dimers form on specific regions of the DNA strand, it can block the activity of DNA and RNA polymerases, thereby inactivating DNA replication and transcription. This eventually leads to cell death. I decided to use UVA in this investigation as it has the unique property of where bacteria are able to repair damaged DNA via photoreactivation. The first stage of this investigation was developing a methodology. Beginning with plating, for my preliminary trials, I concluded that spread plating was the best method of plating, and therefore I would need an inoculum in a liquid medium. So I inoculated LB broth using E. coli K12, which is a non-pathogenic, lab-safe strain of E. coli. I incubated this for 24 hours at 30 degrees Celsius to allow the bacteria to grow and populate the broth. I then performed a serial dilution with the broth to obtain plates with easily countable numbers of colonies. This took a few trials to obtain the optimum dilution, which was 10 to the negative 6, as the images show too few and too many numbers of colonies on some plates. I then inoculated my agar plates with 0.5 centimeters cubed of the diluted inoculum. I altered 2 variables when exposing the E. coli plates to UV light. Firstly, exposure time, and secondly, light intensity by altering distance between the UV lamp and the bacterial plates, each with its own controlled trials. I constructed a large UV light box in which the bacteria would be exposed. After UV exposure, the plates were incubated in an incubator for 24 hours at 30 degrees Celsius before the colonies were counted. Perhaps the most important consideration of this investigation was carrying it out under the aseptic technique to minimize chances of contamination with other microorganisms within the E. coli plates. This consisted of sterilizing surfaces before and after use, using an autoclave to sterilize the broth, agar, and all glassware before use, using a Bunsen burner when inoculating to create an updraft in which airborne microorganisms are swept upwards and away from the work surface. However, despite these measures, some plates showed signs of contamination after incubation. However, these were not included within the dataset. Now the results. I found that the number of counted colonies decreased as UV exposure time increased, as increased exposure time would raise the number of reactive oxygen species formed within the bacterium and therefore increase the likelihood of DNA damage. The number of colonies also decreased as light intensity increased, as increased luminosity means the light is more deeply penetratable to the bacterium and therefore increases the likelihood of forming reactive oxygen species or base modifications. There was no evidence of photoreactivation within this investigation due to the UV light, as the number of colonies shows a linear regression. However, this could be attributed to the fact that it was a short exposure time. While the data has a strong negative and positive correlation, respectively, there are large standard deviations as shown by the error bars, which is a limitation of this investigation. This may be due to the small number of trials, which may lead the data to not be statistically significant. However, the data can be corroborated by other literature studies which found a similar linear relationship between luminosity of UV light and photodamage to E. coli. There are many possible applications of this investigation, such as alternate methods of water treatment, a method of controlling the degree of bacterial disinfection, using UV, and a possible method for damage to skin cells from exposure to tanning beds, for example, which also emit UVA. I've learned a lot from this project, and I hope to continue to do such research that has a positive impact and capabilities of making a difference. Thank you.
Let's now hear out Alexandra San on how she and Paulina came up with their project idea to analyze sleeping patterns.
And congratulations on your presentation. Great that you could make it work to present basically also for your partner who couldn't join you today. Congratulations, first of all. Yeah, we had to ask a few questions on your work. What was the starting point of your study?
Well, me and Paula were like very close friends, and we always talked about also about how sometimes we went to sleep too late, or we were feeling tired during school, and then also in the school we noticed that students look very tired and complain about the lack of sleep. So we decided to investigate further and to find out how big the problem was, and why students actually did not get enough sleep, and what were the consequences of that.
Sounds good. Did you have surprising findings during your work on the project?
Well, it was a bit surprising that students actually admitted that because of their irregular sleeping habits, they had difficulties falling asleep. So they actually noticed that there was a problem. And that was very surprising because usually when you have a problem and you notice it, you try to solve it somehow. So I think that our research can really help the students because we also like put some solutions in at the end.
Is there any recommendations you can give to students now after your studies, or maybe to our general audience with relation to their sleeping habits?
Yes, so firstly they should really try to get the recommended amount of sleep. There's always, you can find on the internet, there's a tabelle, a table which shows which age group needs how much sleep in hours. Of course, that's just a recommendation, but it's still science. So for the general audience, I'm sure that helps. Also, it's very important to not only get that amount of sleep, but have a set bed and wake time, because our body really needs regular sleeping and regular waking times. And thirdly, our third point was that that they should make a routine, a bedtime routine, where they really avoid using their devices, because those devices they emit blue light, and that is stimulating. So it makes it harder to fall asleep, even if you want to.
And there's also things such as filters for blue light, and might also help. And also for glasses, there's also filters, and I'm guessing it's similar for also sports activities, that you should avoid doing those late.
Well, it depends on what sports, because of course, like running very hard or like any stimulating sport activities, yes. But for example, like yoga, that's very relaxing. So we did not study exactly that, but you can really expect that it would not hurt before falling asleep, yeah. But yeah, you noticed I have a UV filter, or?
No, I didn't notice, but I am aware of it.
Do you have it in your glasses?
Yes, I do.
Do you have any? Do you consider your project now as finished and rounded up, or do you feel the need to look further into following the topics, or looking into additional research now?
Well, during our study, we were noticing that more and more, like the more we were researching into it, the more we found could go even further. So it's still like open. We just like began it, because there's really so much more where you can, like, for example, effect on school performance, to investigate further into that. Or investigate further whether it's like the sleep quality, how that is, because there's really a lot of topics where you can still investigate further on that.
So do you already have in mind that you will continue the project and maybe come again next year?
I hope, because actually it's our last year of high school, but and of course we'll go to different universities, but it would be nice to come back. It would be nice, and with Paula as well, yeah. It's nice to have a team partner.
You're having 2 years of experience now in the competition. So is there anything, this is the question we really like to ask to the participants, anything you can advise to people interested in taking part in the competition?
Yeah, so start planning early and just go for it. Because actually it seems so like much work. It is a lot of work, but in the end, like you get recompensated. You can present it. It's not that bad. Even like the jury, it's nice. so really, yeah. So you really, it's like you can only win. Like you not only like win a prize, but you can only like grow also, grow like your. Internally, and develop important skills like making a presentation, writing a research paper, and also to have the Durchsetzungskraft, persistence. Yeah. No. To have like the persistence, because you really practice starting a project and then pulling it through, like really practice this skill, which is really important. Yeah.
So overall, a good experience.
Yeah, really nice.
Good.
Brilliant. Nothing to add to those final words, I believe. So
thank you very much for the interview, and thank you for your time. Congratulations for your work so far, and good luck for tomorrow.
Thank you.
Our next contestants are Amelia Hatch and Ella Pham from the International School of Luxembourg. They will present their project called Food Fraud, Adulterated Milk in Luxembourg. The question they will seek to answer is, are there discrepancies in the safety of milk commonly found in local stores? And what are the potential effects on consumers? I'm excited to hear the answer. Let's hear it from them right now.
Hi, I'm Amelia.
And I'm Ella.
And for our investigation, we looked into food fraud, specifically milk adulteration in Luxembourg. So food fraud occurs when a food supplier adds undisclosed substances to a product, either out of negligence, malice, or economic greed. It is a global issue and has serious effects on consumers' health. We decided to investigate this topic not only because we are very interested in it, but because we believe consumers are not adequately aware of the dangers it poses. Although there is a variety of legislation and regulations in Luxembourg to prevent this from happening, it is still a prevalent problem. In fact, it accounts for 8 to 12 billion euros of damages yearly. A specific example of this occurred in 2013 when horse meat was found in frozen foods advertised to contain 100% beef. Although this may not seem like a harmful substance to add, many people have a cultural aversion to eating it. Even substances added such as sugar can have detrimental effects for diabetics if they're unknowingly consuming it. For these reasons, it is very important for food to be highly regulated in order to keep consumers safe. So we decided to test milk because it is one of the most commonly adulterated foods and substances which can be added to it are formalin, urea, starch, and soap. And these substances can be very harmful to people's health when consumed daily, such as in milk. Before starting our investigation, we realized that it was likely most of our tests would come back negative due to Luxembourg's safe regulations. However, we still decided to test milk because it is so frequently consumed by many. Through further research, we also realized that the substances added to milk are one of the most harmful to consumers. So formalin is one of the adulterants which can be added to milk, and it's added to prolong its shelf life. However, it can corrode and irritate the digestive system, and in extreme cases, it can lead to cancer. Urea can occur naturally in milk, however, producers can also add it in order to increase the solid non-fat percentage, a way of testing the quality of milk. If consumed in large quantities, it can overburden and damage the kidneys. Starch is added for similar reasons, and while it may not seem as toxic to consumers, prolonged consumption can cause diarrhea, and it can also have serious complications for diabetics because it increases their blood glucose levels above a safe range. And lastly, soap is added to milk in order to improve its appearance and give it a thick, foamy consistency, and can cause gastrointestinal complications such as gastritis or inflammation of the intestine.
So our first steps were developing an investigation into adulterated milk in Luxembourg. We started by collecting 12 different milk brands found locally in grocery stores around the country. And then we got into developing different chemical tests to look for all of these different adulterations that can happen. We use sulfuric acid to test for formalin. We use urease and bromethanol blue solution to test for urea. We use iodine to test for starch, and phenolphthalein indicator to test for soap. While we were carrying out these investigations, we paid high attention to health and safety requirements and also our ethical and environmental burdens. We used gloves, goggles, and anti-burn procedures to keep us safe while we carried out these chemical tests and to limit our waste and to limit the negative effect that we had on the environment. We donated the large amounts of leftover milk to our science lab at our school and utilized materials that we had nearby. So, as my partner said earlier, we had already hypothesized that many of the results from these tests would be negative, a sign of the high standards and vigilance that the European Union has for consumable goods for us living in Europe. In concluding our investigation, we found that was mostly true. In testing for all of these different substances, we found that in a large quantity of the different milks, there was the presence of urea. So to confirm these results, we carried out a second trial to test for urea and found that of the 12 brands, brand number 2 and 12 were consistently positive, and brands number 4 and 11 were consistently negative. In spite of these good results and the relative consistency of it, we were still unhappy with the accuracy of our investigation. We found that it was corrupted by the qualitative nature of all of these tests that we carried out and the limited number of trials that we had. In an ideal world, we would have a better investigation by employing DNA methods to test for adulterations in milk and perhaps other foods. We would use PCR and DNA sequencing to test for these for a better accuracy, and we would have more trials. Despite the lack of confidence we may have in the inherent accuracy of our tests, we feel that it's a good representation of the safety that we have in our consumable goods here in Luxembourg and in Europe. Taking it forward, we would want to improve our investigation in the ways that we said before, because food fraud is something that's relatively unknown and an undetected global phenomenon that affects the health and safety of everyone. This phenomenon of food fraud and food adulteration has only been increased by the pandemic, which makes it harder for governments and regulatory bodies to screen and search for substandard food products. In all, we would really want to help move forward and help protect the health and safety of everyone, not only in Luxembourg and in Europe, but worldwide. Thank you very much.
It's now time for a short break, but this will be a special break, as we now have the pleasure of welcoming Oke on stage. Oke is a young artist from Luxembourg who writes and co-produces his own music. After leaving Nigeria in 2008, he settled in Luxembourg with his family and delved into making music with friends. Let's discover his musical universe right now.
Ladies and gentlemen, I'm Oke, and yeah, I'm here to entertain you. My first song is gonna be Sunshine in My Face. It's a very special song, and yeah, enjoy. Come on, clap your hands, come on. Ask me how I came so far, my life spectacular, taught by my grandmother, life is a lesson, no, and why I laugh so hard, that you can disregard, my happiness and fun, which I'm expressing, no, although my family lives across the sea, I always guarantee they stand behind me, oh, and I don't disagree, I've been a refugee, but hey, I found my key, unlock my blessings, oh, when I'm passing by, hey, hey, you see the sunshine in my face, it's my style and my smile, hey, hey, the definition of my grace, that sunshine in my face, come on, clap your hands, face, hey, come on, hey, 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 that sunshine in my face, hey, 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 no, yeah, 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 yeah. And all the time I spend in class, and I just let it pass, picturing how I dance with pride in my heart. They keep asking me, no, they won't believe all my energy, and I can never stop thinking about my homeland, the celebration, the different nations, oh I feel so rich, no more dark frustration, I keep my face up to the sun, life is the greatest gift, when I'm passing by, hey, hey, you see the sunshine in my face, it's my style and my smile, hey, hey, the definition of my grace, that sunshine in my face. Come on, hey, hey, clap, clap, hey, 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 that sunshine in my face, hey, 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 no, yeah, 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 hey, hey, hey. Now I keep my smile, now I keep my smile, yeah, yeah, yeah. Now I keep my smile, yeah, yeah, yeah. When I'm passing by, hey, hey, you see the sunshine in my face. Hey, hey, the definition of my grace. That sunshine in my face, hey, 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 hey. Come on, clap your hands, dance. Hey, hey, that sunshine in my face, hey, hey, hey. Oh, yeah, 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 hey, 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 that sunshine in my face. Thank you very much. Thank you, thank you, thank you, thank you, thank you, thank you.
Thank you, Oke, for this amazing musical experience. Now, let's go back to Aisha Goenka to hear more about her ideas and the research behind her project.
Welcome.
Thank you.
Thank you very much for your earlier talk, and good to be here with you to ask a few more questions on your project. What was the starting point for your studies?
I came across an article about the E. coli infestations in water in Bangladesh in specific. And it really opened my eyes because I think living in Luxembourg, we often take clean water for granted. And we are not aware of the difficulties that other developing nations may be facing with their access to water. And so this made me think of possible ways to solve this problem. And with so much COVID-19 in the news and so many bacterial disinfection methods, I came across ultraviolet light as a form of bacterial disinfection. And so I decided to continue on this path. As microbiology was also a new topic for me, I decided this would be the perfect opportunity to do something new and do some research which has some real-life applications.
Sounds interesting. During your project, have you come across surprising things, or have you faced any specific difficulties?
I think developing the methodology in itself was the biggest difficulty I had. As I said before, microbiology is a new field for me, so when developing the methodology, I had to do some research, and there are so many guidelines, articles, rules in research about how to go about doing this. So navigating that was a little overwhelming, but I thought that the best way to learn was by doing. So I conducted a lot of preliminary trials and to finally see what worked, what didn't, and finally after 6 or 7 trials, I developed a methodology that I was happy with.
Impressive.
And looking further, now that you've participated 2 times, you said, 5 years ago, and now again, with your experience and impressions you could gain during that presentation and studies, what would you tell young students, any advice for them to move ahead with the participation?
I think when I participated 5 years back, it was a very basic project, and I just wanted to participate, come together with like-minded people who were passionate about science like I was. And so I think the best advice I can give is just to take the plunge, even if it's something you don't think is worthy of being presented, or if you think it's basic, chances are it is not. That you know your research better than anybody, that's why you think that way. And I think just having the opportunity in this forum to share your research, your innovations, and also learn from what other people are doing, it inspires you and also just stimulates creativity for what may come in the future.
Sure. And this is quite some significant findings you've had now. Do you have any connection points to transmit your findings anywhere else than in this context of the competition?
This is something that I submitted for my school assignment as well. Yeah. But other than that, I think this is the perfect place to share my ideas. There was such a wonderful jury made up of so many specialists who were able to guide me on what I could do in the future as well. So I think this is the perfect forum to share my findings with other students and everyone who comes to the forum.
Brilliant. And now talking about the future, what's next for you?
I hope to continue this investigation in some regard, whether that be doing it so that I can have more concrete findings or changing some aspect of the investigation to find new material, new investigations, new research. But I think I want to continue down this path of looking at ultraviolet light.
Very interesting.
And also in future, do you already have plans for what you want to study later? Do you want to study in a science-related field?
Yes, absolutely. I hope to study medicine in the future. And I think this is so closely tied with medicine, where prevention is better than treatment. If we can find methods of preventing diseases in the first place, it makes our lives as doctors easier. So I think it's very closely tied to medicine, and it's something that I think I can continue on in my higher university education as well.
Brilliant. Thank you very much for taking the time. Congratulations for your participation and the best of luck for tomorrow also.
Thank you.
Yeah.
Thank you.
See you again.
The next project to be presented on stage is called Which Qualities Make a Lipstick the Best One?, developed by Ekaterina Stolnaya from the St. George's International School. In her project, Ekaterina decided to get as many opinions as possible in determining what kind of qualities people are looking for when picking out their lipstick, which is why she conducted a survey amongst her schoolmates.
I'm sure at least one of you ever had this problem when you try a lipstick on and it's a pretty good one, but it doesn't work. And you think, why is it not the one impeccable one? And that's why I decided to do this project. So my primary objective was to find out which factors are the most important one in a lipstick. And I knew that it's a lot of different factors, like how hydrating it is, color, and many other factors. Then it evolved into finding out what is lipstick actually made from. Because, to reach these qualities, you needed certain ingredients to have. Then I decided to adjust this recipe and find my own one, which will work for me. And finally, I made the lipstick because I knew that it's going to be only mine which will work. Firstly, I made the research, finding out different factors, which I already talked about. But then I understood that there is a huge variety of different factors, so I can't focus on all of them. So that's why I carried out the research at my school for students and teachers to answer which quality is the most important for them in a lipstick. And I will talk about it later a little bit. But it helped me to find out which factor I will mostly work on. Then I adjusted the recipe depending on the factor I wanted my lipstick to have, which after helped me to make the lipstick and eventually try it on and see if my recipe worked. That's my survey, which basically showed that most of the people find that color is the most important factor in a lipstick. And I can agree with it, because for you, it might be pink. For other person, it's red. So I understood that, I won't really focus on this factor, because, it's really personal, and it varies between different people. But the next factor, hydratiness, is the one I really wanted to focus on. The reason I don't usually wear lipstick, because it's dehydrating my lips. I don't feel comfortable wearing it. So that's why I decided really to focus on this factor, to work on it, and make the lipstick which will be hydrating. The lipstick is, of course, made from, general ingredients. It's wax, and I use the bee wax, which provides the shape of lipstick. Then it's oil, which provides the right texture, and I use the almond oil and the silicone oil. Silicone oil also makes the lipstick long-lasting. Then it's pigment, which provides the color, and you can always choose it what you want to use. But I use the food coloring, because it's not only safe, it's much easier to use than beetroot powder, for example. Then, of course, it's the emollients, which provide the moisture of lipstick, and for which, of course, I focused on the most, because I wanted to make my lipstick hydrating. So, what actually makes a lipstick hydrating? And, of course, I use these ingredients. It's vitamin E. So the good factor in it, it's not only making the lipstick hydrating, it also helps to beat new cells, skin cells, so your lips actually become even more softer. So that's why I use a little amount of it, not to affect your health by it too. Cocoa butter and shea butter, they are the best ingredients you can use to make something hydrating, because they're really rich in fats. So the fat acids, they actually build a barrier on your lips to stop your lips using the moisture and hydratiness of them. And, I used all these ingredients, which are not usually used in making lipstick, to make it even better. That's the lipstick I made, and it's actually here. And I was really happy with my results. I spent a long time working on it, because, when you're doing an experiment, it doesn't work from the first time. But eventually, if you put the right ingredients in right measures, you will get a lipstick, a real one, and it's, it was hydrating. All these ingredients actually worked, and it made my lipstick really hydrating. So you don't feel it as something like, sticky on your lips. It's much more gentle, and you enjoy just wearing it. It feels a little bit like a lip balm, and that's what I really wanted to achieve, that you don't feel this awful difference between wearing something which will soften your lips and won't. And this experiment showed me that you can make anything you want and achieve your result by using some simple ingredients. That's the lipstick, and it's really hydrating, and I'm really thankful for this competition that I could actually make something new which would help someone. Thank you.
Remember Amelia Hatch and Ella Pham from before the break? Let's have a quick chat to get to know them better.
Welcome. So, after your presentation, we will ask you a couple of questions to get to know more about your project and you in person. First of all, to start, how did you get the idea to start your project?
I don't know, we were just researching ideas and we came across food fraud and it sounded really interesting and an issue we hadn't heard about before, so we just wanted to investigate it further.
Were there like surprises which you came across during your work on the project?
Yeah, we found some statistics that were surprising. We thought it was more of an issue in other countries, not so much Europe, but realized that it is, and it's hard to control.
Can you give examples?
Yeah, so especially since COVID, it's been harder because for officials to supervise what comes in and out of the country. So they found lots of food that's been smuggled or adulterated.
Yeah, I think in 2020 there was about 320 tons of smuggled dairy products just in the EU alone. So the extent of it is quite surprising. Yeah.
Looking into the topic in a bit of a broader way now, milk is one thing, but generally, do you think that processed food that we get in the supermarkets, is that sustainable and healthy enough? Now, having looked into the topic in more detail?
Especially here in Europe, in the EU, I think it's pretty much safe wherever you go. When you start to go into countries that don't have as strict or as vigilant rules about the agri-food industry and how foods are processed and distributed, then I think it's a bit more worrying, the extent of the problem. But for here, I think it's quite safe.
And then when working as a team, the two of you, did that go smoothly? Was that a good experience for you?
Yeah. I think it's hard with two people to find the times to schedule experiments in the lab at school, but otherwise
I think it worked out pretty well.
Did you distribute the roles, as in one doing a specific type of work and the other one doing another type of work, or did you both do more or less the same?
I think we both helped out with everything, but she likes the science a little bit more, and I like doing the research and reading a little bit more, so we kind of distributed about that way.
And then in terms of doing the actual experiment, there were multiple tests performed, so we each kind of took two.
Looking into your personal career or future projects, would that be an area you'd like to further explore in the future or also consider for your studies?
Yeah, I mean, personally, I'm really interested in the government and law, and I'm heading off to university next year, so hopefully I can take this with me and learn more about it.
Yeah.
Yeah, I think I'm definitely interested in biology. I'm more interested in medicine, and so how food affects humans' health is a very important topic.
Great. Then, yeah, we thank you for now. And we wish you all the best then for tomorrow. Congratulations again for your presentation, your study, and thank you.
Thank you very much. Thank you for your time.
Now, we'd like to welcome the 36th project of a National Jonk Fuerscher contest. Coming from St. George's International School, we have Sebastian Rowe, who will present his project called Does the Improper Storage of Aspirin Compromise Its Potency? Let's find out shortly.
Good afternoon. My name is Sebastian Rowe, and like I said, well, they said, for my project, I chose to do, does the improper storage of aspirin compromise its potency? So, I had to actually find a way to, I had to find a method. So I built this. It's called a corrector. But why does it work at what it does? Well, it's due to something called the Beer-Lambert law. And what that states is that the light absorbance of a solution is directly proportional to the concentration of the attenuating species. What this means is that a specific kind of molecule or species is reducing or attenuating the amount of light that can pass through it. What this means is that more light passing through equals a lower concentration of whatever you're testing for. And in my case specifically, more light intensity equals less degradation. And then I've got this fancy animation. So basically, at a low concentration, a lot of light passes through, but at a high one, less light passes through. It can be written mathematically as the overall light is equivalent to passed light plus absorbed light. This is my corrector. So onto the actual experiment then. Aspirin breaks down with water to create 2-hydroxybenzoic acid. It takes a stupidly long time. So the same product can be recreated more hastily by using mixing aspirin and sodium hydroxide. Adding iron 3 chloride will turn this colorless, undetectable solution purple, and obviously this new purple solution can be detected by my corrector, and using new aspirin, I can make a calibration graph so that my experiment actually works. So here they are, and obviously that's how it works. I stored 2 types of aspirin named Fast Tabs, which is a coated tablet that's designed for quick release, and Aspirin 100, which is a Dutch brand that is uncoated. I stored them in 4 different locations for around 90 days. These were the warm, moist bathroom, the variable car, the hot, dry radiator, and outside, which has just loads of factors, so it's quite interesting. Each location had 3 tablets of each variety to prevent anomalies. So here it is. The remains of the tablets were dissolved in water, giving this colorless, not this colorless solution, but a colorless solution. So again, iron 3 chloride was added so that it turned purple. And so this again can be tested by the corrector, and light intensity was tested, and it was measured in the corrector. So using the fancy calibration graph that I showed you earlier, we could test the aspirin. We could test how much degradation it had. So what are my results then? Well, for aspirin 100, decent results. The bathroom was definitely the best off, which shocked me a little because it's very moist in the bathroom. So I expected there to be a lot of degradation, but it was actually between 5 and 10%, which is quite low comparatively. Outside and the car, they didn't fare too well. However, they were a lot better than the other tablet that I picked called Fast Tabs. Both outside and the bathroom were completely dissolved, which is absolutely terrible for an experiment. In the radiator, there was high levels of degradation, over 35% and up to 60.7. And in the car, there was only a trace amount remaining, so you can't really test it. So how would I evaluate this? Well, there were a few things. Although the corrector behaved according to the Beer-Lambert law, and the graph was produced with really high accuracy, with 97% and 98% accuracy, with the R-squared value, light interference was a problem because, as you can see, there's nothing preventing light from outside getting in, so the lighting in the room would change everything, and even the angle. The cuvette size was a massive problem because it could shift and obviously the light could refract, giving a different result than what we want. Checking on tablets was a problem because, as we saw, 2 of the tablets for fast tabs were completely dissolved. And using pure aspirin was an interesting one because aspirin that I used to compare has bulking agent and other compounds. The problem with this is it's very difficult to get pure aspirin for multiple reasons, even in an actual lab. Oh, there it points out the size. I also would have tested with different drugs, such as ibuprofen and paracetamol. I also got a suggestion that I should use iodine pills, as they're said to have a shelf life of 10 years, which is interesting because in the case of a nuclear fallout, we need to know that we can actually use it and not die. So, how did I fix these problems then? Well, I built this. It looks a lot more professional than the prior design, and everything is hooked up. It's a lot more user-friendly. It just works better. As you can see, there's a housing right here. In fact, there's a housing right here, which... Yeah, there's a housing right there, and obviously right there, which meant that the cuvette couldn't move, as you can just put it on and shift it into position, which is fantastic for me. And that housing also has the additional advantage that light can't get in from outside, as obviously it's a closed-top housing. So that was really useful. So, does the improper storage of aspirin compromise its potency? Yes, but the reason we need to know this is because 2-hydroxybenzoic acid, common name salicylic acid, is actually its own pharmaceutical in itself. It's actually used to remove the top layer of the skin, often used as an acne treatment. However, in high doses, such as the ones you could get from heavily degraded aspirin, then it can actually cause intoxication, causing dangerous decisions and a delirious mind.
Do you want to get some more insights? Do you want to get some more insights into Ekaterina Stolnia's lipstick research? Well, then stay tuned for her interview.
Welcome, Ekaterina.
Thank you.
Thanks for being here with us. And jumping right into the topic, how long did it take you to create your perfect lipstick?
Actually, it took me a lot of time because first, of course, your first experiment won't work. Something will happen. But it actually took me, I think, 3 days. First day was really unsuccessful. And then on the second day, I improved it, put the right measurements. So in the end, I think on the third day, it was perfect.
Oh, wow. Just 3 days?
Yeah.
Oh, wow.
But you need a lot of research before doing it. So yeah, the research took much more time than the actual experiment. Yeah.
And at first, how did you come to the idea of researching into lipsticks?
First, I decided I want to do this competition, and then I thought, what can I do? What can I relate to? And then I started thinking about, people wearing makeup and me not wearing makeup. So I thought, why am I not wearing it? And it was about a lipstick that I didn't really like how it touches up. So I thought, okay, I want to work on it because I have something to say about it.
Is the beauty industry something you would consider also to work in the future?
Partly, yes. No, I wasn't really focusing on really working on it after. But I'm actually more interested in hospitality. But I think, these can actually help me maybe in the future, this project, yeah.
And do you already know if you want to study something science-related?
I will think about it, but mostly no. But I really enjoy sciences and the experiments part the most. So but I don't think my future will be related to it.
May I ask what it's going to be related to then?
Oh yeah, it's going to be 100% related to hospitality, providing people with service, yeah. But this is actually, science is actually also interesting, you know, but I'm more passionate about more mathematical, financial parts.
How did your environment reacted to your project? What was the feedback you got from your findings?
Everyone was really excited, even sometimes more than me. And actually, everyone really liked how it touches up, how it works. So yeah, I think everyone was really happy with my results.
Good then. Any advice you can give to students who consider participating here in Jonk Fuerscher?
Oh yeah, you should definitely do what you're really passionate about, not something you think will work for the audience and the jury, because when you enjoy something, you make it, it makes you much simpler to work on it and enjoy every moment.
Wise words.
Brilliant. Yeah, thank you very much for giving us some more insights into the topic and your approach to creating a kind of improved and perfect lipstick. And yeah, congratulations again. Thank you for now.
Thank you.
And good luck for tomorrow then.
Yeah, thank you.
Welcome on stage our next participant, Alp Eren Baibes from the International School of Luxembourg. Alp Eren is participating with his project called The Effects of Different Synthesis Methods and Catalysts on Crude Aspirin. He will show us to what extent the methods and catalysts used in aspirin synthesis influence the yield, purity, and environmental consequences of the crude aspirin produced.
Hello, everybody. My name is Alp, and today, as they said, I'll be presenting the effects of different synthesis methods and catalysts on crude aspirin. Crude aspirin is just what I used to describe aspirin that hasn't been recrystallized yet. So just a little bit of background. Aspirin is also known as acetyl salicylic acid, and it's produced by adding an acetyl group to the salicylic acid compound at around between 60 to 80 degrees Celsius usually. And it's also usually catalyzed by either sulfuric or phosphoric acid at usual synthesis factories. For this project, I really kind of wanted to focus on the green chemistry aspects of aspirin synthesis. So there are 12 main principles of green chemistry, but I wanted to focus on safer synthesis for the scientist conducting it, increased environmental friendliness, and decreased energy consumption. So for my first targeted outcome, I decided to go with finding a viable catalyst that is basically it produces around the same percentage yield and is also pure enough alongside with the same, for example, with sulfuric and phosphoric catalysts. And my second targeted outcome was to prove that using a microwave instead of a water bath to heat up the synthesis reaction procedure would also provide pretty good results. So I chose 5 catalysts, with one of them being no catalyst at all. The first one was sulfuric acid and the second one was phosphoric acid. I chose these two because they're usually the orthodox catalysts used to catalyze this synthesis reaction. And the main comparison that I would make is in between these same two catalysts between themselves and their difference in the water bath and the microwave methods. And then I also chose propanoic acid as a catalyst. I thought it'd be very interesting because it's an organic acid, it's very biodegradable, and one of the side products produced as a result of producing a crude aspirin is a different organic acid. I also did aluminum nitrate monohydrate as a catalyst because it's solid, so it decreases the potential risk that could be caused to the conductor of the synthesis reaction by a lot. And I also did no catalyst at all to kind of have a ground zero that I could compare all of the different ones to. So to synthesize my crude aspirin, just in a test tube, I added 2 grams of salicylic acid, or around 2 grams. And then I added 3 drops of all of the liquid catalysts, and for the aluminum nitrate monohydrate, in between 0.03 grams and 0.04 grams. And then I added the 2 centimeters cubed of acetic anhydride in the same test tube. Of course, since some of these chemicals are very nasty and very dangerous, I took all the required protective measures. So with the water bath method, after I added in all of the reactants required, I left it in the 80 degrees Celsius water bath for 20 minutes. And for the microwave method, I microwaved it for 3 minutes at the 75% setting with a microwave that had a power rating of 700 watts. And I tested all of my trials. I had 30 trials in total for their yield, their purity with the melting point analysis, and ferric chloride, just to prove that there was still some phenol left in the crude aspirin, to prove that salicylic acid is still in there. And also their environmental consequences, so energy consumption. Just some really quick qualitative observations. All the trials that I produced, except the ones that used aluminum nitrate monohydrate, were white, but the ones that I used aluminum nitrate monohydrate on were kind of a light brown, orangey color, clearly pointing towards a impurity present in there, because aspirin is supposed to be white. And just a quick second qualitative observation was that there was still some salicylic acid left in the weighing boat that I used after I did all of the trials, which clearly pointed that I had a positive skew on the recorded data that I had for the amount of salicylic acid that I added in, which, since all of them had that, it doesn't really matter as much in this case. These are my quantitative results, just to present them in a numerative format. I think it's much nicer looking at them on a graph. So here we can see all of the different trials that I ran and all of their values that I recorded for average mass of crude aspirin produced with different methods, and the average percentage yield, the average melting point range, and the average difference between the commercial aspirin and the aspirin that I made using microwave and water bath methods. So, for the conclusion that I came to on catalysts, with the microwave method, the purest or the narrowest melting point range was present on the microwave trial with no catalyst used at all. And it also had a quite small, one of the smallest, I think, differences from the commercial aspirin melting point range. The best from the green chemistry perspective also is this trial, the microwave method and no catalyst use trial, because even though it provides the purest trial, it's much safer because there's no concentrated acid used as a catalyst, and less energy is used actually due to the microwave. I'll be coming up on that soon. So yeah, decreased risk of synthesis and decreased chemical usage. As an alternative to one of the orthodox catalysts, for example, the phosphoric acid one, we can see that propanoic acid has actually pretty similar results in most of the categories, so I thought that could also be a good viable alternative. As for the microwave method, I compared the energy usage of both of the methods. So for the microwave method, it consumed much less energy than the water bath method at 472.5 kilojoules, whereas the water bath used around 714.78 kilojoules. I calculated these values for the microwave by multiplying the amount of seconds the machine was turned on by its power rating, which was 700 watts. And for the water bath, I calculated by using the specific heat capacity formula of water. It should be kept in mind that the value for the water bath that I obtained doesn't include the amount of energy used to actually keep the water at 80 degrees Celsius. So it should actually be a little bit higher. So for my evaluation, there were some spillages that led to the loss of some trials, which I think is a shame, because it would have provided extra data that I would have been able to then use to create more representative conclusions. And during my melting point analysis of the trials, one of the apparatuses that I used was the lens on it was fogged up, so it made it quite difficult to see when the trial exactly melted. So I think that could have also been a deciding factor. And the biggest evaluation that I had for this project was that I didn't recrystallize the aspirin that I produced, and that it was just crude aspirin. If I conducted these or ran these experiments on the recrystallized trials, I think that it would have been able to hold much more medical and, in general, more meaning as a research. Thank you.
It's always very interesting to hear from our Jonk Fuerscher this year. Let's have a quick chat with Sebastian Rowe to get to know him better.
Welcome, Sebastian. After your presentation, we're going to ask you a couple of more questions to learn more about you and your project as well. To start off, maybe just how did you get the idea for your project?
Well, there were a couple things. I'm very passionate about science and I'm very much planning to pursue a career in the field. So when I was researching available pharmaceuticals to use for my experiment, I thought that maybe aspirin would be a good idea because it's very commonly used and it's also incredibly easy to buy over the counter, unlike some other pharmaceuticals. And then, obviously, after that, I wondered if it degraded, because obviously, if your pills are degraded, they're not going to work as well, are they?
And so are you interested specifically in, I'm guessing, chemistry, biology maybe, or more generally?
Generally medicine and generally just science, but I'm planning to become a doctor. I am interested in pharmaceutics. It's not quite my favourite part of science, but it's still a very, very fun part. It's got a lot of interesting things, like, for example, the whole reason I used iron 3 chloride was to test for the phenol group, which salicylic acid is part of.
You sound passionate in any case.
You can see that clearly. Maybe a question which might be interesting for our audience. So you answered your research question regarding does the improper storage of aspirin has an effect on its potency with yes. So in simple words, how should we best store aspirin or any kind of other pharmaceuticals, medicine at home?
Going off my discussion with the jury, I would very much advise to use the original blister packs because they are quite well engineered to stop air passage into the pills and obviously moisture as well. They're quite moisture and airtight.
Brilliant. Good then. Was there any surprising factors you discovered during your research? Or obstacles, let's say, which you've seen during your research?
I think that there are a few things. I think that sometimes I use the circuit wrong. Like 1 or 2 times at the start, a battery just died on us, which was incredibly upsetting because we had to wait a week for a new one at least, which was quite annoying because it meant we couldn't test as well.
Yeah. Looking ahead on future career paths, what is up for you looking ahead in the future?
I'm planning to become probably a radiologist, but I'm interested in pretty much all medicine. The type of doctor that I'd be least likely to become is probably a surgeon, just because my hands shake a little bit, which obviously isn't a problem with this experiment, which is very good.
Brilliant.
Do you have any advice or tips to other people who would like to participate in the Jonk Fuerscher contest?
Don't get too worried. You're not going to do badly. If you do get too worried, just breathe and try not to hurry too much, or you'll start stuttering and then get worried. So try not to do that, but just relax.
During the presentation, I'm guessing? All right.
Very well.
Thank you very much for your time, your presentation. Congratulations for the work so far.
Thank you.
And I already wish you good luck for tomorrow then.
Cool.
In their project, called Aide Chaussettes, Jan Podolan from the École Primaire Kirchberg and Maciej Goraj from the European School 1 designed an invention aiming at giving people with no hands some autonomy back. Let them show you how.
Hello, my name is Maciej Godaj. I'm 13 years old and I'm a pupil of Polish section in European School in Kirchberg. And I am going to present in English and my friend in Luxembourgish.
Moin, mijn naam is Jan Podolan. Ik ben 12 jaar oud en ik ken een grondvolkje.
Today we would like to present you our project called Useful Sock. We will tell you what it is, for who we created it, how it works, how much time it took us to make it, and what problems we had when we were building our prototype.
Wel 450 miljoen Leute leven in de Europese Unie. Het wordt geschätzt, dat 1 van 5 Leute in de Europese Unie een vorm van behinderung heeft. Het is ganz schwer, die genaue Zahl von den Leuten mit einer Behinderung zu finden. Mit mir all sind Leute mit dieser Krankheit rundherum uns. Für viele Leute haben nicht eine Hilfe bei und können sich keine Sozialhilfe leisten. Laut den Informationen, die wir im Internet finden, wird alle 2 behinderte Personen in der Europäischen Union keine Arbeit. Wir müssen noch den ganz gefährlichen und aufdürfenden Coronavirus aufholen. Wenn es die Pandemie und viele Leute gehen krank, als sie gestorben. Während der Pandemie haben wir Quarantänen. Und viele Leute, die Sozialhilfe brauchen, haben keinen Zugang dazu, weil sie krank oder sterben können.
It's very hard to be independent without hands. People with disabilities depend on the help of other people. Our invention aims to help people without hands regain their autonomy.
Die viele Apparate am Markt, die den Leuten helfen, Strümpfe umzutun. Leider haben sie alle ein Problem. Sie brauchen alle Hände. Es ist ganz traurig, dass kein und die Leute mit einer Behinderung gedurft wird.
So we had this original idea which will allow handicapped people to put their socks on their feet without using hands. Our project will change the future of people with disabilities. So the useful sock was designed as a wooden support. Please see here our prototype. With 2 metal hooks on each side. An integral part of our invention is also a sock, with a zipper in the back and 2 metal circles on each side. At the beginning, we thought we could use magnets instead of holes, but our first results were not very satisfactory. The idea with magnets didn't work because the magnets were too weak. So we tried with metal hooks and metal circles, and it worked. All these different tests with hooks and magnets took us 3 weeks, and we spent 1 month writing the PowerPoint and project paper.
Für Strümpfe umzutun, müssen sie auf den Halter legen und den Fuß einfach rammen. Für sie zuzumachen, müssen sie aus dem Halter rausholen und der Floss muss auf den Haken gemacht gehen. Für sie auszutun, muss auf den Haken gelöst gehen und der Fuß muss einfach herabgezogen gehen. Es gibt den Moment, wo der behinderte Mensch Hilfe von einer anderen Person braucht. Die Person präpariert Strümpfe den Tag für dran. Den Tag darauf geht die behinderte Person Strümpfe einfach an, ohne seine Hände zu benutzen und ist komplett unabhängig von seinem Leben.
And now we will show you how it works. As you could see, it needs a bit of work, as you could see, it needs a bit of practice, but it works. Thank you for your attention.
Merci für Ihre Aufmerksamkeit.
It is Alp Aaron Baby's turn to tell us more about him and his motivations, which got him into science and research.
Welcome, Alp. So after your presentation, I'll just ask you a couple questions to know a bit more about you, your project, etc. Maybe to start off, first of all, what was the motivation, or where did the idea come from for your project?
So I was searching for something to do for my extended essay for the IB, and I chose chemistry because it's one of the sciences and I'm interested. One of the two sciences that I do as a part of my course, and I thought it'd be really interesting to do something with a chemical compound. And since I'm interested in pharmaceuticals and drugs kind of that affect people in good or bad ways, I thought it'd be really interesting to do a research on like a medicine or something. So I investigated and looked over some aspirin research papers, and I thought that investigating the effects of two different synthesis methods, microwave and water bath, with two different energy consumptions could be really good. And I also investigated different catalysts as well, because I found that on the internet or on literature, there were some papers that investigated the effects of different catalysts, and there were some that tested microwave method versus the water bath method, but there wasn't a single paper that combined the two and together and that saw how the different catalysts acted differently in the different synthesis methods. So I thought, why not do that? So.
And how come the choice specifically of aspirin?
Because I find that it's a really interesting medicine. It's one of the most widely used worldwide. It's anti-inflammatory. It reduces pain. You can basically, it's like a miracle drug. You can use it for anything. And it also thins down your blood. And also some researchers find that, has found that it can actually decrease your risk of heart attacks and cancer in some certain areas of your body. So I was like, man, this is pretty cool.
Wow, that's impressive.
So.
During your research, were there any major obstacles you had to overcome?
During my research, my preliminary research, not really, no. But during my actual conduction of the experiment, there were tons of little obstacles that I had to overcome. One of them being the time limit, because I was doing this as my extended essay. At school, we were only given 2 full days to conduct and be done with everything.
Yeah.
So it was pretty difficult and stressful at that time. Yeah.
Maybe linked to that, how much time did you spend on this project?
Just the experimental conduction part. It was around like 8 or 9 hours in 2 days. And then there was also a bunch of preliminary research, of course, and then I wrote it over the summer break because that's what the school instructed us to do, and that also took quite a lot of time.
Yeah, I can imagine.
And linked to the results you have gained out of the study, is there any parts where you want to move forward or explore further in the research with that specific topic?
One of the jury members said something very interesting that I think could be further investigated in this topic, is that catalysts actually decrease the amount of time it takes for the equilibrium in the reaction of the aspirin synthesis. It decreases the time it takes to reach the equilibrium. But what I did was I used the same amount of time for all of the different trials of the catalysts, which kind of doesn't make as much sense if you're purely investigating the effects of the catalysts. But since I was investigating how the different catalysts under the 2 different methods acted, I thought that was the way to go. But what he said, I think, was really important, and I think if there was like a thing to investigate after my research project, I'd probably do that. And that's pretty interesting in this topic.
Yeah, it was great to hear that there is such an intense exchange between the experts and the participants. I think that's most valuable in this contest also. Good to hear that. Okay, and then looking ahead for you personally, what's upcoming? What are you looking into next? And what do you look into potential studies for the future?
Right now, I don't really have one in mind. Maybe as a part of a future project in university. Perhaps that could, then I'll think about it when the time comes, I think.
Yeah, so you mentioned medicine would be one of the areas you'd like to look into as a study option then. Yeah.
Sounds good.
Yeah.
Thank you for the time to answer our questions. Congratulations for your work.
Thank you.
And I already wish you good luck for tomorrow. Hope you get a good prize.
Thank you.
And yeah.
Yeah. Cool.
The 39th project of this year's contest comes from the International School. Developed by Glenn Patch, the project is titled, "To what extent is there a relationship between phosphorus and nitrogen concentrations and excessive cyanobacteria growth in simulated Upper Sur Lake conditions?" In short, Glenn analyzed the effect of nitrogen and phosphorus on the growth of cyanobacteria in the Upper Sur Lake.
Hello, everyone. And yes, that is a pretty long research question. So let's start by breaking it down. So cyanobacteria, which is also known as blue-green algae, is a family of photosynthetic bacteria that, when growing excessively, will start forming what we could call a bloom, simply because there is so much bacteria at the surface of the water that it almost appears as algae, as we can see in Figure 1. And this, in turn, will lead to a decrease in phytoplankton diversity because cyanobacteria produces a toxin that is hugely poisonous to organisms both within and outside the water body if they swam, drank, or even ate an organism from the water body affected. And so in Luxembourg, we have now cyanobacteria occurrence almost every summer. It lasts longer. It starts earlier, and you've probably already heard of it. It is what causes the sort of annual water activity bans on the Upper Sur Lake. And so I was interested in what caused such water bans, and after some research, two possibilities really came. Either climate change or agricultural input, especially nitrogen and phosphorus, leaking from the fertilizers used in crops down into the water body. But the research that was conducted was always ran in conditions that simply could not be applied to Luxembourg because, as we know, we don't have 16 hours of sunlight and 30-degree weather every day for weeks, which would be very nice, but also no rain, which wouldn't be as convenient. And so weather just seemed to have such a great impact that I really wanted to simulate the conditions found at the Upper Sur Lake to really find what was the cause, hence why I researched the impact of nitrogen and phosphorus on cyanobacteria growth in Luxembourgish climate. So to measure growth, I'll simply look at the change in light transmission by measuring it before and at the end of the practical using a colorimeter set at 490 nanometers. Well, I'll alter the nitrogen and phosphorus concentrations in 5 ranges. So no nutrients added, only lake water, then 1, 2, 3, and 4 times the concentration of SARS culture solution, a standard preparation made to grow cultures to maturity in laboratories. And then the remaining 3 variables all control photosynthetic growth or division rates, with temperature and light intensity both controlled by leaving all my cultures outside, exposed to the Luxembourgish weather here from June to July. And CO2 concentration by regularly opening the cap on my water bottles, because that way I could keep a cap on and prevent any spillage. And so here's my methodology in detail, but I think I'll come back to it later. That's what the practical looked. So as you can see, I left the water bottles every day exposed, whatever the weather could be. And after 6 weeks, I was able to collect this data. And we can see here that there's been an overall decrease between the start and the end of the practical on all solutions, but the decrease in light transmission is enhanced as we increase nitrogen and phosphorus concentration. But it still looks kind of messy, so I converted the change in light transmission to percentage increase because some solutions started with different amounts of cyanobacteria, and I was then able to draw this graph, figure 7, where we see a pretty clear logarithmic relationship between nitrogen and phosphorus concentration and the growth of cyanobacteria. And this can be explained by literature, because as we increase nitrogen and phosphorus concentration, we have a faster carbohydrate metabolism, meaning that the majority of the carbohydrates formed during photosynthesis can be utilized for growth and until it reaches the genetic growth potential, where it will eventually stabilize this growth, as we can see at higher concentrations. We can also see from figure 7 that even a minuscule amount of nitrogen and phosphorus already yielded huge growth, with, a 460% increase for only 2 micrograms of phosphorus per liter. And so if we look at qualitative data, we can also find some other findings and possible errors. In this case, we had bubbles of gas indicating photosynthesis occurring, thereby growth, when the temperatures were between 17 to 30 degrees. And this again fits other research that showed that at least one of our 5 investigated strains had its optimum temperature between 20 to 34 degrees Celsius. Now, a problem with the observations of bubbles of gas was that we could only really see them when the light intensity was greater than 3,000 lux. To give you an idea, 3,000 lux is a pretty big light intensity. It is twice the brightness of an overcast day that we often had these past few weeks. And this seems weird because other scientific literature indicated that one of our investigated strain, Planktothrix, had its optimum temperature its optimum irradiance, between 2,150 to 3,500 lux. So we should see bubbles of gas being produced under 3,000 lux. Now, it could be because Planktothrix is usually found within 4 to 15 meters of depth, and I could only really collect surface water, which shows that there is a bias in this research. Another observation which kind of weirded me out in a way was that we could only observe brilliant cyanobacteria, meaning cyanobacteria at the surface, in the lowest nitrogen and phosphorus concentration, where I added no nutrients. And as I mentioned earlier, as we increase nitrogen and phosphorus concentration, we do increase the metabolism of carbohydrates. Hence, we must have a lowered storage of carbohydrates, making the cyanobacteria lighter and allowing it to float. But it could be due to the SARS culture solutions that I used, where the powder remained undissolved, as we can see in figure 9, even 6 weeks after the practical ended. 6 weeks after the practical started, And so this could hint that in a way nutrients couldn't really reach the upper layers of my water bottles, hence cyanobacteria that started to become buoyant, quickly became nutrient starved, and sank back down. But what's the greater point of this research? Well, we can come to a conclusion that nutrients, so nitrogen and phosphorus especially, were needed to sustain the growth. And they did up until a certain point where it started to plateau. But in order for any growth, for any bloom to occur in the Luxembourgish climate, we do need appropriate light intensity and temperature. We can see then how much of a problem it will become with climate change, because we'll have conditions that are more that are better for cyanobacteria growth, with hotter, sunnier, drier summer. And as we saw here, only a small amount of nitrogen and phosphorus was needed to actually stimulate growth. So we could have cyanobacteria in lots of water bodies, long as that threshold is already reached. It's a really small threshold. So we will quickly start seeing huge impacts on the wildlife, that, affecting directly fishing, but also tourism with the water bans here, as we can see in Luxembourg. And also a problem on the drinking water supplies, which we're not really concerned here in Luxembourg because we have proper treatment, but imagine for the countries that can't treat their water as well as we can. Of course, we could limit the problem by removing nitrogen and phosphorus from the water supplies, but this will reveal costing 3.1 billion euros in the Baltic Sea. Cyanobacteria proliferation may then become a major climate change impact here in Luxembourg, but also in Europe. Thank you.
Up next, we'll hear from Jan Podolan and Maciej Goraj. Let's learn more about how they came up with their ideas and their motivation behind helping people with facing difficulties.
So welcome, Jan and Maciej. Great to welcome you as the youngest participants this year in the contest. And yeah, excited to ask some further questions on your project, which really has relevance for everyday lives of people. So what was the starting point or the inspiration for your project?
Plötzlich, was war die Idee, wo da zu dem Projekt gekommen sind?
Wir waren an der Schule von den Leuten gefragt. Da wollte ihnen helfen.
And how did you like working together on that project?
I don't know how to answer this question.
No problem.
Hast du dann nach Hause gegangen, für zusammen zu schaffen, zu zweit? Im Projekt?
Ich von nicht verstanden.
War es okay, für zu zweit ein Projekt zu schaffen? Oder ist ein bisschen gestritten? Oder?
Nein, voll okay.
Gut eingehen für ein Projekt?
Kann man nicht verlangen.
Do you think that your project can be go on a major scale into proper production actually? Is that something you can think of?
I think that maybe, because it's very helpful.
Yeah.
So a lot of people with disabilities can just buy it and like use it.
Yeah. Yeah, it was great to see how simple the setup actually is and how good the idea can be, and helping people, yeah, as you say, in their everyday lives, having disabilities, and yeah, simple but very efficient tool. So that's very welcome to hear. How was the reaction of your schoolmates on the project? Did you go into discussion on what you are doing? Were they asking questions, curious about that?
No, I didn't talk about it. Because I don't like when somebody just watch me when I'm presenting. So I didn't want my friends just to watch me. So I didn't tell them.
We kept it secret all the time until today. Okay, I think now's the time to share on the live screen and to share the link maybe that your friends can watch it, what you've been doing. How long did you work on your project?
We just meet like one time a month when we were building our prototype.
Yeah.
And when we were writing, we just met. We were meeting one time in a week.
Okay.
And it took us like 3 weeks to write the project paper.
Ja.
Und dafür eventuell das Projekt weiterzumachen oder ein anderes Projekt zu machen, und nächstes Jahr rumzukommen?
Wir wollen nächstes Jahr rumkommen. Diese Probe gibt man vielleicht noch verbessern. Ja, wir wollen nächstes Jahr zurückkommen. Mit dem neuen Projekt.
Ein neuen also. Klingt gut.
Great. I think you have a few more years to come back then. And yeah, thank you again for sharing that prototype with us and your way to cooperate in that project. I think that's a good inspiration for everybody, even at young age, to just go for it, enter the contest and learn a lot, and also for the audience to learn a lot. So thank you very much, and then all the best for tomorrow. Thank you.
Felicitationen für Ihr Projekt und die ganze Arbeit und dann schon viel Glück für morgen mit der Preisüberreichung.
Yes.
Welcome to our three next scientists, Jennifer Mendes-Singh, Anne-Marie Bach, and Lina Benali. They come from the Lycée Classique de Diekirch and will present their project titled La diversité des sols au Luxembourg. Let's hear about their research.
Moin, wir sind Lina, Jennifer und Anne-Marie und wir präsentieren euch heute das Projekt, mit dem wir uns die letzten paar Monate beschäftigt haben, La diversité des sols au Luxembourg.
Habt ihr schon einmal vorgestellt, dass ihr mehr wie nicht mit einem einheitlichen Boden trippelt?
Nein, wahrscheinlich nicht. Aber genauso war es bei uns am Anfang der Fall.
Genauso wie die meisten haben wir, wenn wir und die Natur gedacht haben, immer nicht mehr das gedacht, was ihr gesagt, wenn ihr richtig ausguckt. Leider machen sich die Monster großartige Gedanken darüber, was die Source von dem Ganzen und von vielen weiteren Phänomenen ist, nämlich der Boden.
Ja, voilà. Aber genau das wollen wir ändern. Weil wir haben nämlich die Chance, an einem Land zu leben, wo es immens viele verschiedene Böden gibt, auf kleiner Fläche.
Du viel mehr als auf sich gemacht, und wir haben 4 Böden im Gesicht und haben denen ihre Charakteristiken in der Sicht auf Vergleich. Wie das aber ein immens großes Thema ist, mehr als nicht mehr auf die abiotischen Faktoren beschränkt.
Wie schon erwähnt, gibt es viele verschiedene Typen von Böden hier im Land, von denen wir als 4 ausgesicht haben. Wir sind zum Beispiel an Eisle gegangen, an Fjels, auf Meistreff und auf Kiespelt, an Hundo, ständige Boden, sandige Schluffboden, sandige Boden und schwere Lehmboden fand. Am Ganzen gibt es da 24 hier im Land.
Mit den 4 verschiedenen Böden haben wir nur 6 Experimente gefordert. Für mehr über die Proprietäten, die wir nur absuchen werden, und noch über die Unterschiede herauszufinden.
Uns war es wichtig, dass wir alles selber entdecken und ausprobieren. Dafür haben wir zum Beispiel die Möglichkeit aufgelehnt, als Echantillon einfach am Labor aufzugehen, für das sie die Arbeit für uns machen.
Als erstes Experiment haben wir Couleur du Sol geguckt, das heißt Farbe vom Boden, mit der Hilfe von diesem Munzel Color Chart. Ja, hier ist es. Und wie ihr auch heute vielleicht guckt, oder wir können den noch Mino weisen, also du hast schon einen scharfen Kontrast mit der Farbe von den Boden. Dann haben wir die Textur du Sol geguckt, also oder so genannt Granulometrie, oder aus was dem Boden besteht. Leider hat man nicht technische Mittel, für es genau festzustellen, also haben wir uns und oberflächliche Eigenschaften beschränkt und auf die Bodenkarte.
Als nächstes haben wir uns für die Struktur des Bodens interessiert. Das heißt, ob der Boden eher krimmelig oder kompakt ist. Dafür haben wir mit einer Schippe den Boden abgeschlagen und haben dann in Hand die Form von den verschiedenen Partikeln analysiert. Als nächstes haben wir uns dann auch auf den Humusgehalt vom Boden dafür interessiert. Und das heißt, wie viel organische Materie am Boden dran ist. Wie das die Materie brennbar ist und der Rest vom Boden nicht, haben wir dann den Boden verbrannt, für dazu bestimmen, wie viel Humus am Boden dran ist.
Wie ihr gesehen als die 5. Experience Kapazität de Retention en eau. Dafür haben wir mit einem Rohr an den Boden geschlagen und geguckt, wie viel Wasser all Boden abholen kann. Als nächstes und als letzte Experience haben wir nach der Acidität du Sol bestimmt, also den pH-Wert von all verschiedenen Typen von Boden, die wir geholt haben.
Wir haben immens viele verschiedene Resultate für die verschiedenen Böden gefunden. Und das ist wirklich erstaunlich für uns, wie viele verschiedene Böden wir hier im Land haben. Wir konnten auch anhand von unseren Resultaten als viele Phänomene hier im Land, wie zum Beispiel die Inundationen, besser erklären. Das ist faszinant, wie die Klangfaktoren vom Boden so viel an unserer Umwelt beeinflussen.
Genau. Und wie gesagt, haben wir 4 Bodentypen geguckt und analysiert. Und wir konnten ganz verschiedene Resultate. Jeder vielleicht am Project Paper oder viel gesehen hat. Ja, und wie Lina viel gesagt hat, gibt es da 24 Typen von Boden. Stellt euch vor, wir hatten sie alle geguckt, dass wir noch mehr viel Unterschied. Und das ist nämlich Beweis, wie groß das Thematik eigentlich ist, und wie viel du von der Sorge kann. Wir haben zum Beispiel gar nicht von den Organismen am Boden geschwärzt. Wir fanden, dass das Thematik ganz wenig Aufmerksamkeit kriegt, ja, weil wir wenig da konfrontiert gehen.
Wir haben ehrlich gesagt auch immens lang gebraucht, für auf das Thematik warten zu kommen. Aber gerade du, wenn es leidet das wirklich um Herz, dass wir die Leute dafür inspirieren können, sich für die Thematik zu interessieren. Es gibt am Anfang schon relativ viele Recherchen hier im Land gemacht bezüglich zu der Thematik, zum Beispiel von List oder von der Osta. Aber es gibt am Anfang viel zu wenig darüber geschwatzt.
Dafür hoffen wir, dass wir mit einer kleinen Präsentation von nimmen 6 Minuten davon erzählen konnten, was auch unser Hauptziel ist von dem Projekt.
Vielmals merci für die Nullstelle.
Let's now welcome back Glenn Parch to learn more about his motivation and ideas.
Welcome, Glenn. After your presentation on your project, we'll just ask you a couple more questions to learn more about you and your project. Maybe to start off, what has been the trigger, the idea to start this project?
Yeah, so I mean, when I first went to the Upper Sewer Lake, it was during a school trip, and we were supposed to have like water activities, etc. And it was just all cancelled, and that was the first time I really learned about cyanobacteria. And then I came to 11th grade, where we needed to essentially have a scientific project, and I really wanted to see a climate change impact here in Luxembourg. And it didn't really occur to me like the first time, but then I realized, like, oh, whenever I would read about the water bans in the Upper Sewer Lake, they would always mention kind of climate change as one of the causes. And I just thought, why not? Why not like carry on with the research? And yeah.
And following that project, is environmental science something you'd like to pursue also for future projects or even for studies afterwards?
Yeah, so I don't think it'll be directly environmental science, but it will definitely be something related to climate change mitigation. Mm-hmm. So next year I will study in part molecular biology to kind of look and hopefully go into biotechnology later on.
Were there things you discovered during your work that surprised you or that you didn't expect at first?
It was definitely just how few nutrients could actually yield like that much growth, you know. It was kind of a shock to find that actually you could have like a toxic amount of cyanobacteria growing in, like, for example, Evian water. If you had the conditions needed, for example, good weather, that was appropriate for that. And yeah, that was definitely something. And these conditions will again come with climate change, etc. But don't worry about it. You don't have cyanobacteria in your water, in your Evian water. If you were to add it, yeah, it should grow to a good amount. But don't worry about it at the moment. We're good.
During your project, did you face specific difficulties while going about it, or not really?
Yeah, I mean, the first 2 weeks I wouldn't have any growth at all. And yeah, that was also something I was really surprised about, was actually the light intensity needed. Because it just wouldn't grow whenever I would like put them inside or etc. I would really need to have strong sunlight outside and leave them for a full day to actually see that growth happening. So yeah.
Okay. After your participation in the contest, is there any advice regarding finding the right project or approaching a project you can give to anyone interested in Jonk Fuerscher for the future?
I think that definitely you should do something you're really interested in. Because, I mean, for example, mine, it like took almost all the summer holidays. And sometimes, again, like the 2 weeks where it wouldn't grow, it was really, I kind of want to say demoralizing. It was like, I know this is supposed to work, it just doesn't. And so I think you should really have that passion, that interest to really always be flexible, find new ideas and new ways to actually go through with your project and then present it on stage. And again, I think if you're interested in the project you're presenting about, your interests will directly be like visible on stage, etc., and will help you.
So follow through and try again and again.
Yeah. Yeah.
Sounds good.
Yeah, I think it's interesting to hear that many participants do mention those points. First, do what you like, because then you can be passionate about what you're doing. And second, keep on going, whatever it takes, even though you might need to overcome some obstacles on the way. But yeah. Yeah.
Well, thank you very much for answering our questions.
Thank you.
And congratulations for the participation in the contest. And yeah, just going to wish you good luck for tomorrow for the prize ceremony.
Yeah, thank you.
Thank you so much.
Last but not least, our final participants of the day are Francine Aben and Lynn Schneider. They both come from the Lycée des Redanges and will present their project called Bioplastique à base d'amidon. The aim of this project is to have a biodegradable plastic made from potatoes.
Bonjour, je m'appelle Francine.
Et je m'appelle Lynn. Nous allons vous présenter notre projet bioplastique à base d'amidon.
Les images ci-contre, qu'ont-elles en commun? Elles représentent toutes un des problèmes mondiaux actuels, celui de la surconsommation et de la surproduction de déchets, dont un des plus néfastes est le plastique, qui a un impact néfaste sur la biodiversité. De plus, ceci concerne également le Luxembourg.
Voilà pourquoi nous nous sommes focalisés sur notre propre région, qui semble touchée par le littering. Voici donc des photos que j'ai prises en promenant mon chien, qui adore ramener des bouts de plastique qu'il trouve par terre. Or, aussi cette collecte du plastique ne va pas résoudre le problème en soi. Le plastique ne se décompose pas. C'est la raison pour laquelle nous nous sommes intéressés au bioplastique.
Or, la dénomination bioplastique n'est pas un terme protégé. Cependant, elle nous fait parfois croire que les produits bioplastiques sont issus de produits agricoles biologiques qui sont écologiques ou bien compostables, mais ceci n'est pas nécessairement le cas. Un bioplastique peut être soit biosourcé, à base de matière végétale par exemple, mais pas forcément dégradable. Et puis, il existe également les bioplastiques biodégradables, qui peuvent également être à base de matières fossiles. Nous voulions donc créer un bioplastique biodégradable à base d'amidon, par exemple sous forme d'une housse de smartphone ou d'un film alimentaire. Alors le modèle idéal serait d'avoir une économie circulaire. Ça commence avec la production agricole des pommes de terre qui peuvent être transformées en industrie, en fécule. À partir de cette fécule, on pourra créer une résine bioplastique pour obtenir notre produit final, le bioplastique, qui pourra être décomposé pour à nouveau créer de nouveaux produits agricoles. Mais pour avoir vraiment le modèle idéal, il faudrait quand même un produit agricole qui n'entre pas en concurrence avec la production alimentaire. Donc, nous avons pensé aux pommes de terre parce que l'amidon peut être récupéré des déchets de l'épluchage des pommes de terre.
Nous avons donc commencé par l'extraction de la fécule. On a épluché et râpé des pommes de terre et après filtration répétée, on a finalement obtenu de la fécule.
Pour déterminer la pureté de notre amidon, nous l'avons comparé à de l'amidon industriel. Et ici, vous voyez très bien qu'en fait, sous le microscope, la coloration de notre amidon est beaucoup plus foncée que celle de la fécule industrielle, où nous avons également trouvé des pelures de pommes de terre. Donc, notre fécule était plus pure.
Par la suite, nous avons chauffé tous les ingrédients nécessaires dans une casserole jusqu'à l'obtention d'une pâte gélatineuse que vous voyez ici. En variant les rapports entre les différents ingrédients, notre produit final a obtenu de différentes structures et rigidités. Voici donc une fois du bioplastique coloré en vert, un premier prototype d'un film alimentaire, mais celui-ci est encore un peu plus friable, et un deuxième film alimentaire plus étirable qui fait rappeler le film plastique alimentaire traditionnel. Mais que s'est-il passé en chauffant tous ces composants ? Une réaction de polymérisation a eu lieu. Lors de celle-ci, chaque composant occupe un rôle particulier. Ainsi, la glycérine et notre plastifiant donnent structure et rigidité au plastique. On nécessite un acide, le vinaigre, et de l'eau, qui est le solvant pour notre polymère, la fécule de pomme de terre.
Nous nous sommes ensuite intéressés à la réaction des acides avec notre bioplastique et on a trouvé qu'en fait, il a vraiment réagi avec l'acide sulfurique de très haute concentration. Donc, on peut conclure que notre bioplastique est plutôt résistant face aux acides les plus courants. Pour savoir si notre bioplastique est biodégradable, nous l'avons trempé dans de l'eau où il s'est complètement dissous et après 3 semaines dans de la terre, il s'est dégradé en de tout petits morceaux, donc il est compostable.
Nous avons également essayé de créer un autre type de bioplastique, le PLA, ce qui signifie polylactic acid. Ce plastique est donc à base de l'acide lactique qui peut être synthétisé par l'amidon. On a donc chauffé un tube à essai auquel nous avons ajouté du chlorure d'étain et de l'acide lactique afin d'obtenir une pièce de bioplastique sous cette forme. Mais il faut quand même remarquer que la production de ce plastique est beaucoup plus difficile et qu'il nous était sûrement possible de créer une petite quantité à la fois.
Pour conclure, nous sommes parvenus à créer un film alimentaire à base d'amidon, mais pas vraiment à créer de petits objets. Nous sommes convaincus que les bioplastiques biosourcés et compostables sont une solution possible pour le problème du plastique.
Idéalement, on consomme moins le plastique en général, mais il serait assez difficile d'abandonner complètement le plastique. Avec une amélioration de sa production, notre bioplastique aura une empreinte écologique positive et pourra ainsi s'imposer comme solution à long terme. Merci pour votre attention.
Before ending this year's presentation part of the contest, we would like to hear more from Jennifer Mendes-Singh, Anne-Marie Bach and Lina Benali, who will then be followed by Francine Abben and Lynn Schneider from the Bioplastic Project.
Moin Anne-Marie, Lina und Jennifer. Nach eurer Präsentation haben wir noch ein paar Fragen über die und ihr Projekt. Ich will mal anfangen, einfach, wie Sie dann die Idee kommen auf Ihr Projekt?
Wir waren schon immer relativ bezogen in der Natur, zum Beispiel ich sind immens viel an der Natur oder zu Fuß oder mit Pferd, ich weiß nicht. Und voilà, mir schon immer am Anfang, habe ich immer gesehen, also am Anfang hat man am Anfang Thematik mehr zum Borkenkäfer, weil wir haben immer gemerkt, dass die Büscher Fuzzi sind am Anfang. Und danach hatte mein Gespräch bei den Vierstadt, der hat gesagt, dass das ganz am Anfang einfach den Boden bezogen ist. Und wir haben allgemein, wir haben relativ viel Problematik genug geguckt und immer mehr gemerkt, dass der Boden Source von dem Ganzen war. Und voilà, da haben wir da immer mehr so eine Recherche dran gebracht und ich fand schon mal wirklich spannend, dass Boden die interessanteste Thematik für uns ist. Ja,
genau.
Während der Recherche sind da auf Sachen gestolpert, wo ihr euch überrascht habt oder wo da dann Schwierigkeiten hat?
Alors, je parle en français. Du coup, oui, bien sûr, il y a eu beaucoup de difficultés, surtout le fait qu'on n'a jamais eu de cours d'écologie, par exemple, à l'école, sur ces thèmes-là. Donc, vraiment, c'était un sujet qu'on a dû vraiment chercher. Donc, même dans, je ne sais pas si vous avez remarqué, mais un de nos slides de la présentation se nomme « En quête du thème », parce que c'était vraiment une quête pour nous de trouver un thème qui nous ressemblait et avec lequel on s'amuserait quand même à rechercher tout ça. Et donc oui, il y a eu beaucoup de difficultés, surtout le fait que on n'a pas vraiment donc eu de cours et que il y avait des expériences où on n'avait pas le résultat qu'il fallait, donc on devait en refaire. Et ça, c'est un peu les aléas de la science, mais ça nous a toujours plu et on a toujours continué à le faire.
Ja. Da kann noch kein so fügen, was war wirklich ein Challenge für uns, weil an der Schule auch wenn wir da TP machen, was wir relativ oft machen bei unserer Aktion. Da kriegt ihr immer gesagt, voilà, ich möchte den da und wir hoffen dafür, auf das Resultat zu kommen. Und heute war es wirklich so, wir müssten selber auf die Idee kommen, okay, was wollen wir erreichen und wie kommen wir da hin? Und das war wirklich ein Challenge für uns, mir tut das auch viel beigebracht.
Nee, so ein Challenge frei halt eben.
Ja, voilà.
Und ich muss nicht vergessen, dass auch die Pandemie da mitspielt. So, das ist auch oft, dass man als wir haben uns nur geteamst, was auch gut, aber so war umständlich, uns immer zu treffen. Wir wohnen noch weit von den Nähen, dann ist das auch umständlich.
Ja.
Und, ja.
Et à cela s'ajoute ma question. À travailler à trois en équipe, comment vous avez approché le sujet? Est-ce que vous avez eu des responsabilités ou des tâches prédéfinies? Qui fait quoi? Ou ça s'est développé au cours du projet?
Je pense que ça, on a un peu fait au feeling, enfin on a un peu fait à notre manière. On n'a jamais dit écoute toi tu fais ça, moi je fais ça, parce que c'est pas vraiment comme ça qu'on fonctionnait. On s'est plus entraidé à faire toutes les tâches ensemble, mais bien sûr qu'il y avait certaines choses où on ne peut pas perdre du temps à le faire à trois, donc chacun devait s'occuper de quelque chose de spécifique. Mais c'est vrai que tout au long du projet, on s'est toutes les trois épaulées parce qu'on se connaît quand même depuis assez longtemps et qu'on est dans la même classe. Donc non, ça va, on a quand même su se partager les tâches sans pour autant faire un travail individuel.
Mir noch alle verschiedene Fähigkeiten, die man dann noch alle zusammenbräuchten, zum Beispiel Lommerte Sprachen, also typisch Lützeburg. Ich sehe mehr so der Deutsch, Lützeburg, hat kann jemand gut Französisch und hat kann jemand gut Englisch. Und da tun also auch wirklich viel Brüche, dass man wirklich alle gut verschiedene Fähigkeiten haben. Voilà.
Ja. Gehen jetzt Sachen in Ihrem Projekt für der Welt weiterschaffen oder eventuell ein anderes Projekt machen und für dann nächstes Jahr Konkurs mitzumachen?
Aber so weit haben wir noch nicht gedurcht. So, wann, ich weiß nicht. Am Punkt doch schon.
Ja, also mir hat es definitiv möglich geht, weil mir als wie ich direkt am Anfang so mir also die abiotischen Faktoren beschränkt. Mir natürlich die Boden, du hast auch immens viel Biologie dran, du sind Insekten, voilà, wie immer mal gemacht, du sind auch ganz viele Bakterien oder Champignons, die da mitspielen. Und das Ganze, das ist eine immens interessante Thematik. Wir haben das alles auf der Seite gelöst, wie das eine immens große Thematik, also kann ich nicht alles machen. Aber dafür definitiv etwas, wo ihr drüber weiter schaffen könnt.
Ja, das ist ganz wahr, weil ganz am Anfang waren wir, oh, mich Schatz von den Champignons, mich Schatz von den Organismen, mich Schatz von der Heu. Und dann nur, nee, wir müssen alles erschrecken.
Ja, genau.
Okay. Ok, bon, on vous remercie beaucoup d'avoir pris le temps de répondre à plusieurs questions. Félicitations encore pour le projet. Bonne chance pour demain.
Ah oui, merci.
Et on sera content de donner l'opportunité au public aussi de faire une expérience de votre projet demain. Donc, merci encore.
Merci à vous. Merci.
Ja, willkommen. Vielen Dank für den Vortrag. Es war wirklich sehr interessant. Als erste Frage wird uns wirklich interessieren, wie seid ihr auf euer Thema gekommen?
Also wir haben in der Schule so eine wissenschaftliche Option, heißt das hier in Luxemburg. Und wir haben nach einem Projekt gesucht, da unser vorheriges Projekt schiefgegangen ist. Und dann ist, glaube ich, Franz und ich durch Recherchen dann auf diesen Bioplastik gekommen. Und dann haben wir verschiedene Arten, haben wir uns reingelesen, was sich am besten eignet, was sich auch mit unseren beschränkten Mitteln irgendwie durchführen lässt. Ja. Und sind eben dann auf diesen Bioplastik gekommen.
Seid ihr während eurem Projekt oder du Sachen herausgefunden, die euch überrascht haben?
Ich meine, wir sehen, dass der Tisch ein paar Mal schief gegangen ist. Natürlich, wir hatten da ein paar Rückschritte, wo wir dann noch gucken mussten, wie wir irgendwas darüber hinwegkommen. Und ich meine, das war am Anfang das Schwierigste und das war das am meisten überrascht wird.
Jetzt als Folge eurer Recherchen habt ihr Themen, die ihr weiter behandeln möchtet, wo ihr nochmal tiefer einsteigen möchtet, und könnt ihr euch auch vorstellen, vielleicht nächstes Jahr nochmal wieder teilzunehmen mit einem Folgethema oder einem ganz anderen Thema?
Ja, absolut ist es, weil et Kinder sich mehr Film hier gestaltet und das probiert, wenn er nicht wirklich so Objekte oder so, das heißt, vielleicht mehr in die Richtung noch zu gehen oder dann auch eben zu gucken. Von der Jury hat gesagt, den ökonomischen Teil, das ganz mit den Bioplastiken am Anfang noch nicht immens rentabel ist oder so. Und eben zu gucken, inwiefern da Möglichkeiten gehen oder so. Und dann auch mit der Kompostabilität, der Biodegradabilität ein bisschen mehr genau zu gucken.
Ja.
Wo da mit eurem Projekt ein bisschen Recherche gemacht hat, oder Tipps für die Leute an puncto Recycling und so, wo in dort verschiedene Sachen aufpassen muss oder so?
Also ich meine, du können mir Leute selber nichts Neues zählen. Du kannst einfach wirklich die üblichen Sachen, probiert Sachen lassen zu kaufen, machst du kommen viel mehr ab, du kriegst viel mehr Sachen an Plastik zu kaufen. Regionalsachen sind noch mehr einfach ohne Plastik zu kriegen. All die typischen Sachen, Plastikflaschen evitieren, wir haben so viel mittlerweile Gläser und Flaschen, Möglichkeit darüber auszuweichen und einfach viel mehr auf sich selber gucken, wo kann ich Sachen reduzieren, wo kann ich umswitchen auf etwas anderem. Und ich meine, da können mir wirklich die Leute nichts Neues mehr erzählen.
Wirst du da schon an ihrer Studien wird auch so wissenschaftlich related Studien machen?
Wissenschaft nicht zu 100%. Ich meine, ich will Richtung Psychologie gekommen. Aber trotzdem da immer bei deren irgendwelcher Kontakt bleiben. Natürlich, da wäre für mich persgestützte Therapie, wie für mich so den Traum.
Und ich interessiere mich so für Irrsinnsindustrie, also und die ganze Produktion alimentaire, weil ich am Anfang ganz interessant fand. Oder mehr Richtung Medizin oder so, mehr schon etwas mit Naturwissenschaft.
Okay, also schon eine Verbindung zu
ja.
dem, was ihr gemacht habt. Konntet ihr andere Schülerinnen von eurem Projekt auch begeistern? Habt ihr euch ausgetauscht? Wie seid ihr das Thema angegangen, auch Jonk Fuerscher ein bisschen populärer zu machen vielleicht in eurem Umfeld?
Also wir haben uns ja am Anfang noch nicht so wirklich ausgekannt, wir haben noch nie Heimat gemacht, das heißt, wir konnten noch nicht wirklich den anderen erzählen, wie das als heute sind oder so. Aber wir haben ganz eine Science-Option gemacht bei uns am Lycée. Und da sind noch Leute, die sich eben für Science interessieren. Das heißt, wir schon ein bisschen mit denen geschwatzt. Und wir haben noch einen Prof, der uns eben geholfen hat, das ganze Material zur Verfügung zu stellen und uns motiviert hat, für das mitzumachen. Und ich meine, das tut uns auch viel auf die Weg halt gebracht.
Super.
Klingt gut, dass mitgeholfen wird mit Schülern.
Ja. Gut.
Na dann, merci für das Interview, merci für die Antworten. Merci für die ganze Arbeit, die ihr an eurem Projekt gestartet habt. Dann bleibt mir nur viel Glück zu wünschen für meine Preisbereicherung.
Merci. Vielen Dank und viel Erfolg für morgen dann.
Merci.
We have sadly arrived at the end of this year's contest. 43 incredible and innovative projects were presented here on stage, brimming with creativity and the will to improve the future.
A huge congratulations to all of our amazing Jonk Fuerscher. Do remember that you can visit the stands of our participants in person tomorrow as of noon. So please come down to the forum to look at the innovative projects and ask any questions you want to our Jonk Fuerscher. They will be so happy to exchange their ideas and answer your questions.
Tomorrow, we will find out what prizes our participants will take home. And before we say goodbye, please welcome back our great Luxembourg artist, OK, so that we can finish a great day with good music and in a cheerful mood. See you tomorrow.
Thank you very much. My next song is going to be Freedom. It's a very special song, and yeah, as we all know what is going on in Ukraine, to the people of Ukraine, I'm with them, and I wish them freedom. I wish the people in Syria, in the Middle East, in Africa, my home country, Nigeria, I'm sending my freedom wish to them. So this is for them. I'm breathing easy, I'm letting go. And all my guards, they are falling down. It's time to be me, no hiding out. I'm so much more than what you know. My eyes are doors to this world, reprising fault, it still turns. And Lord, I know of its worth. Freedom and no freedom. Freedom. And I'm with you, we are one. Freedom. No freedom, freedom. I'm with you, we are one. Take a look around, you're not alone, you're not alone, yeah, yeah. And no perfection found, but you are loved, oh yeah, yeah, yeah. We are travelers, we share one home. No more prejudice thoughts, we're moving on, new baby. My eyes are doors to this world, reprising faults, it still turns. And Lord, I know of its word, yeah. Freedom, no. With you, we are one. Freedom, freedom, yeah, yeah. Freedom, freedom, freedom. And I'm with you, we are one. Ooh, yeah, 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 yeah. Don't have no time for this. To live with happiness. Just like Christ and humans' rights. Love and freedom, preach. No, yeah, yeah. I got the freedom, freedom, yeah. Woo! I'm with you. I'm with you. We're one. I got the freedom, freedom, yeah. And I'm with you. I'm with you. We're one. Freedom.
with you. I'm with you. We'll no, yeah, yeah. No, yeah, yeah. Hey, freedom, 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 yeah. Now with you, we'll want freedom.
Thank you. Thank you, thank you, thank you so much. And my next song is a very special song. I'm from Nigeria, so Lagos, born and raised. And the next song is called Love is Free. And in our language, Yoruba, we say Ifemi. That is the same thing as love is free. So I think love is a universal language, and everybody understands love. And I'm sending love to you guys at home today. So let's go.
Take a look around. Tell me, come on, clap your hands. People sharing love, why they don't look like me? People sharing thoughts who have different beliefs. So why you keep on telling me that love ain't free? Love is free, yeah. For you and me, love is free. For you and me, love is free. Yeah, me and you and love is free. For you and me, love is free. Yeah, love is free. Love is free. Although my pockets look poor, I keep well in my mind. Need no society to show me what's wrong, what is right. So I'm writing a song, those I want you to feel. Those are wrong would try to tell you that love ain't real, yeah. Love is free. Oh my, yeah, take a look around. Tell me, what do you see? People sharing love, why they don't look like me? People sharing thoughts who have different beliefs. So why you keep on telling me that love ain't free? If and me, my love is free. If and me, love is free. Oh yeah, 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 yeah. For you and me, love. For you and me, love is. Hey, me and you and love is free. For you and me, love is free. Yeah, baby, love is free. Woo. Love is free. Yeah, 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 yeah. If and me, my love is free. If and me, love is. If and me, my love is free. If and me, love is. If and me, my love is free. If and me, love is free.
Thank you very much. Thank you so much. And to all those who want to support me, please feel free. My name is OK Music on Instagram. I'm on Spotify as well. Please follow, like. Thank you so much. Thank you guys.